120 people logging in. Well, that's a record for us. And I think it shows, and just been talking to John and Anastasia, that we should be looking at this type of activity in the future. Certainly, it's a good way of getting a lot of people together, given the costs of travel within Africa, going for even from one country to another. So I think that's certainly something we can look at over the future, and also helps us to have sort of mini conferences in between time on given subjects. I think we started yesterday with an excellent presentation from Andy welcoming us all and obviously giving us a brief history of MURIA, how it started in 2015. We weren't sure then whether it will be successful going forward, but we've certainly shown that just by yesterday and all the activities in between. We were really fortunate yesterday morning to have an excellent presentation from Faith Okolobo, really about medication use in special populations, and really general advice about how to undertake drug utilization studies and look at pertinent indicators. Interesting, that came up in theme 2 yesterday because we had discussions on pictograms, management of diabetes, etc., as well as really discussions building on work in Namibia about maybe it's time to move away from the WHO inward criteria to more, if you like, special ones or ones for Africa, particularly because of the high rate of both infectious and non-infectious diseases. So to discuss less than 2 medicines per patient is probably difficult, where, for instance, in Namibia, I think 40 to 50% of the adults have hypertension and quite a high percentage have diabetes, and obviously patients have infectious diseases on top. So maybe it's time for us to look and develop specific indicators for Southern Africa, and we can build on that. And we had Sean talking to us yesterday and came to Namibia, and my colleague Stephen Campbell in Manchester has helped us out in Namibia, and he was responsible for developing the quality and outcome framework indicators for the United Kingdom, particularly England. And Sean, as you well know from Namibia, is basically in charge of developing quality indicators for Scotland. So I think between those two, maybe we've now got enough data and experience within ambulatory care in Africa to really say, should we start to develop our own indicators? So that's something that we can discuss later on today, and then perhaps with those two go forward with it. We also had some very good examples of COVID-19. We had some good examples of activities in Ghana, also activities in Zambia to develop their own sort of hand sanitizers, etc., as well as proactivity among pharmacists in Namibia. So that shows that really in Africa, we've been doing very well to date and need to build on that. One thing we can look forward to, and maybe bring it up later today, is really the unintended consequences. So if we lock down people with non-communicable diseases, then what happens to their hypertension, diabetes, etc., and also their medicine supply. So maybe that's something we can discuss later on this morning as a way of going forward. We also heard a number of point prevalence studies, as well as studies around antimicrobial use in hospitals. That's very important for us going forward if we're looking to improve antibiotic use and to reduce AMR. So we looked and build on those in the future. And we also had a very good presentation from South Africa on the app that they developed for PPS studies, building on what we developed in Botswana. That's important because if we've got an app that can rapidly collect data and more importantly, provide real-time findings, then that helps us really to develop and implement quality improvement programs quite quickly within hospitals. I think we'll see more of that in the future, so I'm looking forward to that. As I said, we had a number of good presentations on non-communicable diseases, which is a key area for Africa going forward, particularly because of high rates and growing rates of non-communicable diseases. And as we know, really quite poor adherence rates currently for a number of reasons. So if we could look to build on that and pictograms and other things are a way forward, then that helps us in the future. So obviously we'll be looking more at that type of activity in the future. We also heard from Kenya about concerns with warfarin management among public hospitals in Kenya, and then we opened it up wider. And then we also heard about problems really across most of Africa with warfarin management, both in terms of really having the capacity to monitor warfarin, INR levels, etc. So we should look to build on that. Joseph came up with an example in Nigeria, I think, where they start patients off in the acute setting with some of the newer oral anticoagulants like rivaroxaban. So maybe we can look at that going forward, but certainly we need to improve our warfarin management. So that's obviously an area for us going forward. One thing that struck me yesterday, because I moved between the rooms because I'm presenting and listening in both, that we had about 270 at one stage in room 1 with theme 1 and only about 30 in room 2 with theme 2. So if you look at the program, that gives you a link to go forward, to back and forward between the rooms. So just follow that, and that allows you to do it quite quickly. So please move between the rooms if you can. Lastly, before I finish, I'm going to say looking forward to today and later on this morning. And as you see right at the bottom, we've now collated all abstracts. Joseph did that yesterday, and we've now sent that to Ilse, so that will be on the website soon. But before I finish, can all presenters in the plenary room please raise their hand so that we know who you are, ready for John to hook you up. So please, can all presenters in room 1 in the plenary room raise their hands so that John can put you on. John, are you seeing raised hands?
None at the moment.
So
I can see Charles. Huh?
Yeah, Charles is here.
Okay.
So we need Charles is there. Charles, can you raise your hand?
Well, I can't see my hand, so I can't raise it.
Oh, it's on the meeting icon. You
I
have
saw it was there yesterday. It's not that today.
Yes. Oh.
The icon is
Oh,
not.
you are moved. You are ready. You are okay yourself. Charles, you're okay. You are panelist already.
Okay. And what about Fola or Bokwanda?
Okay. No, we can't see. Yes, we can't see him at the moment. Maybe he still, the session, we are 4 minutes ahead of schedule.
Well,
So
that gives
maybe
us 4 minutes. That should be. So
yes.
Fola, are you there? Mr. Nokwanda, are you there? No. Kevin, are you there?
Yeah, Ilse is in room 2.
Yes,
Is in the
yes,
second
I'm
room.
here.
Yeah.
Okay.
So
Francis
Ilse is presenting. She's in room 2.
Yeah, so the rest are in there on that. We had, I think, this morning, I think Languta from South Africa, I think we had an email, Joseph, didn't we, that she was sick?
Yes.
So that allows, if you like,
You shouldn't.
more time so you've got obviously more time
later.
There. Then I'll head on to Fola's probably in room 2. So if I log on to room 2, I can say he needs to go back to room 1 to present. He might be already in room 2.
Yes.
Otherwise, I think that's it really. So we're slightly
I think
ahead.
Harriet is in room 2. She's presenting.
Rachel Issa.
Yeah, Rachel Harriet. Yeah.
So she's already Harriet. So she's so that's fine. They're already there. Right. I think that's it now. So I think we can probably, if I go over to room 2 and tell Fola that he's probably starting off in room 2 and then ready to go into room 1. Right. That's all I've got to say. So I'll quickly see if I can get into room 2 and then.
Raise your hand so that we can be able to allow you to speak. By making your panelist. If you're a presenter, kindly raise your hand.
So Joseph, I leave it to you now. I'll go over to room 2.
Okay. So we're going to.
Yeah, you have about 2 minutes
Okay.
to quarter past, so we can give the 2 minutes so that we start.
Yes.
Hi, Joseph.
But yeah, so I think how are you,
I'm all right.
Francis?
I'm
Cool.
all right.
That's very nice. So how was your night?
I rested.
Yeah, good. So I think we're in Charles as the first presentation.
Yeah.
Yep.
Okay. Olayinka is not yet on today.
He told me he would not be readily available. He'll
Okay.
be
Okay.
caught up at work. So
No problem. No problem. We'll take care of that.
yeah.
So I think you have the floor. You can introduce Charles, and then we can start.
All right. Thank you.
Yes,
Good morning, ladies and gentlemen. You're welcome to today's session. And without further ado, I'd like to introduce Dr. Charles Ezeduka, who's going to present on the antibiotics dispensing practices among community pharmacies in Anambra State, Nigeria. Dr. Ezenduka, the floor is yours. You have 10 minutes to present, after which you will go through a 5-minute session of questions from which we will expect answers from you. The floor is yours. Please go ahead.
Okay, thank you, Francis. Hope you can hear me.
We can hear you.
Thank you. And ladies and gentlemen, welcome to this morning's session. And this is also welcoming, acknowledging Professor Godman. Been so many years we've spoken last. So thank you for this opportunity. And the slides are with you. Hello.
Dr. Ezenduka,
Yes.
will you please share your slides with us?
Okay. Is it there now?
It's coming. I think it's on its way.
Okay.
Okay, there we go. Please
All right.
make it full screen.
Okay. All right.
Okay, and I'm timing you now.
All right. Thank you. So I'll be talking on dispensing and sales of antibiotics in community pharmacies in Anambra State. That was carried out in 2017. And this is a presentation plan. By way of introduction, we know antibiotics are used to treat infections caused by, which are mainly communicable diseases. And this constitutes a huge burden, disease burden in Africa in particular. And they're often categorized as prescription-only medicines, which should be dispensed based on prescription from recognized physicians. But we know that these drugs are obtained without prescription in some drug outlets and community pharmacies around the world. In Nigeria, antibiotics are designated as POMs and should not be sold as over-the-counter medicine. So that pharmacists are supposed to prescribe against valid prescriptions. However, over-the-counter use of antibiotics has resulted to a lot of consequences. Inappropriate use has since become a serious global problem, with multiple adverse consequences: antibiotic resistance, failed treatment outcome, and increased costs, to the patient. Now, community pharmacies represent a major source of antibiotic globally, where over 93% of people obtain their antibiotics. This poses a major problem. This is as a result of poor regulation, self-medication, and economic motives, which encourage the abuse or misuse of antibiotics. Now, this information is important in Anambra State, which is noted for major drug chain distribution around the world, especially in West Africa, due to the popular bridgehead market in the States. So it becomes necessary to assess how this practices is being conducted and then the consequences it poses to the population. So the study was carried out to determine the extent to which antibiotics are being dispensed and sold. Answer questions are: what proportion of antibiotics is dispensed without prescription through the community pharmacies? Do all community pharmacies in the state involved in this practice? What are the factors that influence the sale of the antibiotic without prescription in the community pharmacies? And what are the most commonly dispensed antibiotics among the community pharmacies? So study methodology was cross-sectional survey that was based on the use of semi-structured questionnaires and of course patient simulated client method. All pharmacies registered in the state were included in the study, and all of them cut across the states where you have over 90% of community pharmacies spread in the 4 main cities: the capital city, Onitsha, the major city, Nnewi, Ekulobia, and maybe Nkpo in Anambra State. So of all the 126 officially registered, 96 were made the criteria for the assessment, and the data collection was undertaken in 2017 between June and August of that year. Now, the questionnaire tool was divided into 4 sections to collect relevant information: demography, evaluation of the pharmacist's knowledge about the guideline that inform the use of antibiotics, the attitude towards the use of antibiotics in the community pharmacy, and then evaluate practices on how pharmacists actually dispense antibiotics. And to confirm this practice, we also use the simulated client method, whereby 2 scenarios were created. One for which antibiotic was requested for treatment of adult cough, and then a request for antibiotic for treatment of pediatric diarrhea. So what were the findings? Of the 96 questionnaires that were distributed, 89 was returned correctly filled. More than half of respondents were below the age of 30, ranging between 31 and 40 years. They were equally divided between male and female respondents. Majority of these respondents were community pharmacists. So what were the results? This table shows the knowledge of respondents towards the use of antibiotics, like answering questions about awareness.
5 minutes.
Awareness about the use of antibiotics based on prescriptions, and of course, how the practices were being, how their knowledge of how they are being used all over the community pharmacies in the state. This second table also shows the attitude of pharmacists towards the use of antibiotics without prescriptions, which of course gives us the reason why antibiotics are being prescribed or being dispensed without prescriptions. And that one of which was the confidence that they have the knowledge to use it, and the patients do not want to go to the hospital. But most importantly, economic reasons and pressure from the owner for increased sales and profit. Now this table shows the commonly dispensed antibiotics by class: penicillins, the quinolones, and of course the cephalosporins, these individual drugs. This shows common medications or common medical conditions for which antibiotics are being prescribed in the state. Now on the patient simulated or simulated client method, which was used to confirm the actual practice, showing that majority, if not all of them, actually was involved in dispensing of antibiotic without prescription. So in summary, the extent of sales of and dispensing of antibiotic without prescription in the study outlet shows that almost all the premises or pharmacies were involved in the sale of or dispensing of antibiotics without prescription, 88%. And the most frequently dispensed antibiotics include the penicillins, followed by quinolones, and of course the cephalosporins. 70% were unaware that dispensing antibiotics without prescription is illegal. In the stimulated client method, almost all requests for antibiotics without prescription were granted in both scenarios. Now, economic motive of increasing sales and fear of losing a client or patient were the major reasons for dispensing antibiotics without prescriptions, similar to previous studies. Now, the percentage of pharmacies selling antibiotics without prescription with a licensed pharmacist, whether it is manned by a pharmacist or based on a pharmacy staff, there was no significant differences. There was no significant difference between pharmacies with the pharmacist on duty and those without pharmacists on duty. Now, under the simulated client method, the percentage of antibiotic dispensed on demand with a licensed pharmacist on duty significantly lower, very understandable. Now, penicillins are the most commonly dispensed antibiotic without prescription, presented by amoxicillin and ampicillin, clozacillin. This is consistent with other studies like you have in Indonesia and some other parts of Nigeria. Findings from simulated study scenario shows or confirm the fact that antibiotics can easily be obtained without prescription in the state community pharmacies. So in conclusion, it is clear that over-the-counter sales of antibiotics is a common practice in community pharmacies in Anambra State. Findings also confirm widespread use of antibiotics without prescriptions in medicine retail outlets in Nigeria as in most developing countries. Primary causes being lack of regulatory enforcement and economic motivation. Therefore, our recommendation is strict regulatory enforcement and structured educational campaign to the public on the need for doctors' prescription for effective use of antibiotics. The pharmacy staff also required to be trained regularly to enable them know that it is important that antibiotics are used based on prescriptions. References. Thank you. Hello.
Dr. Ezenduka, for a good presentation. I'd like to invite the attendees to pose questions at this moment. There is a raised hand. Olainka. Dr. Olainka.
I think he has some problem. Maybe he's muted centrally.
Okay.
Hello, Francis.
Okay. Yes.
Oh, good.
Sir.
Oh, okay. Good morning. Sorry, I've been hearing all the conversation. I've been part of it. You couldn't hear me.
Oh, okay.
Okay.
You may
All
ask
right.
your question, Doc.
No. I wasn't asking question. I just wanted to solve the technical problem so that I can.
Oh, okay. All right. Is there anybody with a question? Okay, I just want to ask a question myself. This is, thank you very much for a good study, Dr. Ezenduka. I just want to ask, is there a chance, because if you enforce the law, Nigeria is big, I'm aware, the population is very high, and also time, finances for patients going to doctors for prescriptions, for diagnosis and prescription might not be easy. Is there a possibility that there may be need for training of pharmacists to gain ability to prescribe, that is to do review of the cases for simple ailments like upper respiratory tract infections, simple urinary tract infections, so that they can be able to make decisions that are based on logic and therefore withhold the use of antibiotics from the patients? Thank you.
Yeah, thank you, Francis. Of course, there are, and it's been recommended in the past to make it official, but I don't think it's official. such things, measures are in place. In fact, sometime early this year, programs like that were undertaken by organizations on the need for clinical pharmacists to always be updated on the use. And I can tell you, many of them are competent, and but the law probably has not allowed the use of community pharmacists in managing simple conditions. But again, that will also require a diagnostic test to make sure that you don't use it blindly. Okay, so it is there, but it has not been made formally legal in Nigeria.
Okay, thank you very much. Anybody else with a question?
I can ask a question, Francis.
Please go ahead.
Yeah, Charles, in your presentation, you mentioned two categories of facilities where the antibiotics were handled, and one of them was drug outlets and community pharmacies. So in this particular state where you did your research, how would one be able to distinguish between a drug outlet and a community pharmacy? And are these legally established facilities? So I just don't understand.
Thank you. If you listen very well to the study was done in only community pharmacy. That's why the premises were only 96 registered. The criteria was they had to be registered by the regulatory agency. The other legally established outlets are the patent medicine vendors, which was not part of the study. The study mentioned particularly community pharmacies. Okay, so other study has been done where, of course, we expect, what do you expect if you do such a study in patent medicine? Nigeria regulation is quite poor. So, but it's good in the light. So over there, some studies have been done. I did myself in the past, 2011, where most of those practices are also seen in the drug retail outlets, apart from community pharmacies.
So my next question, thank you. My next question is, when you are doing an assessment of the knowledge level, did you target the knowledge level of pharmacists or even other cadres of personnel that were serving in the pharmacies?
Yes, if you look at the table 1, where we said the knowledge
is
expressed.
Hello?
Yeah, Kennedy.
My question is,
somebody
yes, Kennedy. I'm the one Kennedy talking, huh?
The last person did not conclude. I was answering her.
Okay.
Hello.
Hello.
So have I finished answering your questions?
No. What I didn't see is if you actually assess the level of knowledge of the different levels of cadres of staff working in the pharmacies, did you do an analysis to confirm which cadre had the lowest level of knowledge?
Well, if you look at the table on the knowledge of respondents, see that every respondent was assessed on the level of knowledge, not by way of what do you know, but by expression. And then some of the results showed you that responses from pharmacists shows more understanding of the use of antibiotics. Okay, so the person without prescription was more for the non-pharmacists than the pharmacists themselves. The problem is that oftentimes non-pharmacists are also involved in dispensing without the pharmacists being around. So other studies have been done. It's very clear that pharmacists have more knowledge on the use of antibiotics, but the question is that do they undertake that diagnostic tests to ensure that you don't use the wrong antibiotics that is not sensitive.
Okay, thanks.
Okay, thank you. I'd like to ask Bivale.
No?
Bivale raise her hand before
Hello?
Kennedy.
Okay.
Bivale. Bivale Karume. Okay, Kennedy, you may ask your question.
Okay, fine. My name is Kennedy. Can you hear me?
Yes, Kennedy, please go ahead.
Okay, fine. My question is, according to Dr. Charles, yeah, like, it's like when it comes to pharmacy, they can just do it like antibiotic over the counter. My question is, in Kenya, there are so many restrictions, and in Nigeria, it's like there are no restrictions, especially when it comes to second line antibiotics.
Hello?
Yes.
Did you ask a question? I didn't get that.
Okay, fine. How the restrictions in Nigeria when it comes to over-the-counter antibiotics? Like in Kenya, there are so many restrictions. Somebody must have a prescription.
Well, like I said, the laws are there, but implementation is a problem. Hardly do you see any regulation on the practice in itself. So because of that, people do a lot of things in the premises. So many studies will tell you regulation, enforcement of regulation is a problem in most developing nations, particularly in Nigeria. So there are restrictions, but are they being implemented? Are they being monitored? So that's the issue. The law is clear how this should be carried out in Nigeria.
Okay, thank you, Dr. Ezenduka. I would like to request Dr. Ogunleye to introduce the next speaker.
Not sure if Olianka's there.
All right, thank you very much, Francis. Hello, Brian. Hello, Brian.
Hello, good morning. Carry on. Sorry.
Okay, thank you. All right. So thank you very much, Dr. Zeduka, for the presentation. Our next presenter is Mr. Nokwanda Bilakati, and he's going to be speaking to us on physiochemical assessment of selected brands of artemether-lumefantrine tablets obtained from retail pharmacies in Umbane in Eswatini. Mr. Vilakati, you're welcome. Please, you have the floor. You have 10 minutes, and then we interact with questions and answers for the last 5 minutes. Thank you. Are you there, Mr. Vilakati? We can't hear you. Please, can we hear me?
Okay, let me check. Is it part of the
We're not
panel?
hearing him. We can see his slide.
Let me see if it's there.
He's sharing his slide already, but we can't hear him.
Okay, let me see. Is he included as a panelist? I can't.
That's what I'm trying to check now.
Mr. Amo, are you with us? Because Mr. Amo is included as a panelist, and I think they work together. So
they work together, so you can carry on with it.
Joseph, Afola was supposed to be leading in theme 2, and I've just been over there and he wasn't there. So I'm not sure if he's around.
He's around, okay. Okay, so maybe if we can get it.
Is in the pool of the other attendees? I'm just trying to run through the list.
But then we've
so
got your. But on my screen, I've got you're sharing Fola's screen, so somebody must be doing it.
Yeah. Yeah, it's sharing Folari's screen, yes.
It's sharing the screen, yes.
Yes. So Mr. Nokwanda, with us.
You've got Emu's here. Okay, I've got that. So I say, please.
Emu
Okay.
could present because we made him a panelist. He could have been the one who shared.
But I've just got here, he's just said to all panelists, Emu is here. So I just said, please speak. Hello, Emu. So Fola, are you there?
Okay. So,
hello.
Maybe having some.
He is there. Probably he is either muted or he has not put on his microphone.
Yeah, on that.
Okay, Amu is here. So please can Mr. Folari Amu unmute himself and speak?
He's unmuted, but I think he could be having a network issue because it shows his microphone is not muted.
Yeah.
Or do we move on to the next presenter?
Let's move on.
Yes, in the interest of time, it's probably better to move on to the next slide because he could be having.
We'll take it back later.
Yeah.
Yes.
Okay, so Mr. Muise, may you allow the next person to come after sorting out place. Okay. All right. So we're going to take the next presenter. Okay, just 1 minute. Okay, so why they are sorting the technical issues they are having out, we're going to invite Mr. Kevin Awere, who is going to speak to us on comparative prevalence of hypersensitivity and central nervous system effects of a DTG and an MVP-based regime in HIV patients at the Kenyatta National Hospital. Mr. Kevin Awere, please can you share your slide and make your presentation? You're welcome, Mr. Kevin.
Thank you very much.
All right, great, thank you.
Thank you very much. My name is Kevin Awere from the University of Nairobi in Kenya. We did our study in conjunction with Professor Gwantai and Dr. Oluka, and we looked at the prevalence of hypersensitivity and central nervous system effects in HIV patients on dolutegravir and nevirapine-based regimens at the Kenyatta National Hospital. Now I will do the presentation in the following format. We'll have a brief introduction. Then we look at the objectives, the design and method, the results, the discussion, and then finally the conclusion. Now, as you are all aware, DTG is among the latest additions to the HIV treatment guidelines, and it comes loaded with advantages, one of them being once daily dosing, fewer side effects, and fewer food, drug, and drug-drug interactions. However, it also has some documented adverse drug reactions. Among them is insomnia, headaches, and anxiety. And because of these many advantages, DTG is slowly replacing the traditional ARV drugs, especially those in the non-nucleoside reverse transcriptase inhibitors class, in which nevirapine is one of them. And being a relatively new drug in Africa, its safety is not as well documented as the traditional ARV. And therefore, our objective for this study was to compare the safety of dolutegravir and an nevirapine-based regimen in HIV patients at the Kenyatta National Hospital, specifically with regard to hypersensitivity and central nervous system effects. Now, this was a cross-sectional mixed with a historical cohort study, which had two groups. One group was on the dolutegravir regimen, and the other was on the nevirapine regimen. And for ease of comparison, they all had a common backbone ART of tenofovir, disoproxil, fumarate, and lamivudine. We obtained our data from both physical and electronic medical records. And to augment this, we also did some patient interviews through a structured questionnaire that was administered and filled by the researcher. Now, the study population involved all patients who are on the two regimens from the period of June 2017 to April 2019. Our inclusion criteria. These patients must have been on this regimen for at least 6 months. They must have had a good medication adherence record, which is recorded at every visit at the hospital, and they must have given voluntary informed consent. We used the purposive sampling method to carry out this study. Now, when it comes to analysis, to compare the effects between the two groups, we used the chi-square test, and to test the impact of other variables, we did logistic regression. Our primary outcomes were 5. Under the CNS effects, we looked at the prevalence of insomnia, headaches, and suicidal ideation of tendencies. And under the hypersensitivity, we looked at pruritus and rash. Now, in order to achieve all this, we graded all these effects using the division of age table for grading of severity of adverse events. This is version 2. This is how the table looks like. In the interest of time, I shall not go deeply into it, but basically, we divided all these adverse drug reactions from mild, moderate, severe,
excuse me.
life-threatening.
Yes.
Excuse me, can everyone mute their microphone because we're getting interference? Sorry, Kevin, carry on.
It's okay. And for the hypersensitivity reactions, we used the same table, which was also graded into 4: mild, moderate, severe, and potentially life-threatening. Now, we got 111 participants who met our eligibility criteria. 86 of them are from the DTG arm and 26 on the NVP arm. If you look at the table, there was no statistically significant difference in the ages of our participants across these two groups. However, when it came to the gender and the duration of treatment, there was a statistically significant difference, whereby we had more females than males, and those who were on the nevirapine-based regimen had been on ARVs longer than those who had been on DTG regimen. Now, when we look at headaches, there was a statistically significant difference between these two groups. However, all instances of headaches were either mild or moderate. We did not get any instance of either severe or life-threatening headache. Then insomnia. For insomnia, a majority of them, again, were mild and moderate. Grade 1 is mild, grade 2 is moderate, grade 3 is severe. There was no statistically significant difference, once again, between these two groups. However, in the absence of statistical significance, we had a prevalence of about 4.7% of insomnia among the DTG group, which I believe has some clinical significance. Now, coming to suicidal ideation and tendencies, again, there was no statistical significant difference between these two groups, and a majority of them ranged from mild to moderate, but we did have some instances of severe suicidal ideation or tendencies, with the DTG arm having a slightly higher prevalence of 2% compared to 1% for the TDF, CTC, and nevirapine arm. Coming to pruritus, majority of them again was mild, moderate, but in this particular parameter, there was statistical significance difference, whereby we can see that we had more of mild pruritus in the nevirapine arm of the study than the DTG arm of the study. And we also had a 2% prevalence of pruritus in the nevirapine arm of the study. Now, we did not find any cases of rash in the dolutegravir arm of the study. However, we did find a 4% prevalence of rash in the nevirapine arm of the study. Now, when we tried to question these findings, from literature, we could not find documented evidence associating nevirapine with insomnia. So we did a multivariable logistic regression, and it became apparent that this insomnia was statistically more associated to the sex of the patient rather than the regimen itself. And that has been, as you can see from the p-values. However, our prevalences, especially for the DTG arm of insomnia, mirrors that of other studies that have been done, the single study, but differs from that of the spring study. The single study got a prevalence of 4%, while the spring study had a prevalence of less than 1. And headache, again, was more associated with the duration of treatment, whereby we found those who had been on their regimen longer were more strongly associated with having headaches than those who had been on it for a shorter time.
I'm giving you another one. There's another one I'm quoting from somewhere.
Now, in conclusion, from our study,
How many minutes?
thank you. In conclusion, from our study, we concluded that participants who were on the dolutegravir regimen were significantly less prone to headache and pruritus compared to those on the nevirapine-based regimen, and that there was no significant difference between the two regimens when it came to insomnia, suicidal ideation or tendencies, and rash. Before I finish, I'd like to acknowledge the immense contribution given to me by a number of people. The first ones being the study participants themselves, the clinical and the laboratory staff at the Kenyatta National Hospital, the management itself, and also my co-authors, Professor Gwantai and Dr. Oluka. Thank you very much.
Thank you very much, Kevin, for that brilliant study and presentation. Please, can we all unmute ourselves? There's a lot of disturbance from the background, please. So I'd like to welcome comments and questions from all participants. Please, you can just raise your hand if you have a comment or a question. Okay, Francis, please unmute yourself and speak.
Okay, thank you very much, Kevin, for your presentation. And I see you are into pharmacovigilance or pharmacoepidemiology, drug safety. Kennedy, what is your conclusion in regards to the safety of dolutegravir? I know you had a small sample size, but would you like to say something about what you think about the safety of dolutegravir, and also comment on what is lacking, what else you're going to be looking at for further studies? Thank you.
Thank you very much.
Kavi, you can speak.
Okay, thank you very much. From our conclusion, we concluded that number one, before I get to the conclusion, you realize that our sample size was not as big, and therefore this was more of a pilot study. And from it, we could see that dolutegravir, in terms of these two aspects of CNS effects and hypersensitivity, was no different from the regimen that we have been using in several aspects. And therefore, we concluded that its safety is more or less the same as that of the NNRTI with regard to these two parameters. And in future, we hope to have a larger sample size, number one, for this study, so that our study power will be a little bit higher. Thank you.
Okay, thank you very much. Can we have Vonai Ungotole ask a question?
Can I also make a comment before she comes on board?
Please go ahead, Prof.
I think one of the observations that Kevin should mention is that it was not easy to get many participants on the nevirapine arm because of the government policy to transit the patients that were on nevirapine arm to DTG. So he tried very much to get participants, and they were not available. So he was able to recruit all the patients that were on board during the time of the study.
Okay, thank you. Please, Vonai Ungotole, please can you ask your question or make your comment?
Okay, my question is about the DTG.
Carry on, please.
The dolutegravir, about the safety. What about to the women of childbearing age? Does it not cause the neuronal defects to those ladies? Thank you.
Okay, Kevin, did you hear?
Yes, I did hear that.
Okay, maybe before you answer, so that you just take few comments on the chat too, you take it together because we are running out of time for this slot. So somebody said, does sex mean gender? That's another question. What are the possible co-founders? Okay, is nevirapine-induced headache and pruritus life-threatening? Because considering the conclusion, not much of a difference is seen between DTG regimen and nevirapine. So maybe you speak to those two. Thank you. Kevin, over.
Sorry, I didn't get the last one.
Okay, the last one, it says, is nevirapine-induced headache and pruritus life-threatening? Were they life-threatening? Because considering the conclusion, not much of a difference is seen between the two drugs. He said, was the nevirapine-induced headache and pruritus life-threatening?
Okay, thank you very much.
And any co-founders too? Those are the other questions.
Okay.
Thank you.
To answer the first one, the headaches that we encountered and the pruritus, as I mentioned earlier, were largely mild and moderate. Remember, we graded all these adverse reactions into 4 groups. We had the mild, moderate, severe, and potentially life-threatening. So from our study, we did not get any instances of severe or life-threatening headaches. I think that should answer that. Can you hear me?
Dr. Olainka, you are muted.
So what about Voda's question?
Yes. Voda's question was on potential confounders.
Women of childbearing age.
Or women of childbearing age.
Yes, and risk to the fetus and all that, I guess.
Okay, unfortunately, that is outside the scope of our study because we based our safety on 2 aspects, hypersensitivity and CNS effects. So the study did not assess the teratogenic effects of dolutegravir.
Okay, thank you very much, Kevin. We'll have to move on to the next presentation now. Thank you for the very good study. Others who might be interested in other aspects of the study could interact with him later. So over to you, Dr. Calimera, for the next presenter.
Thank you, Dr. Gunle. I would like to introduce Dr. Godfrey Mutashambara Reguerera, who worked along with Professor Gottman, who will present on alanine transaminase and hemoglobin appearing to predict the occurrence of anti-tuberculosis medication hepatotoxicity.
Like Francis, it will be me, because unfortunately Godfrey is traveling at the moment, so he's listening on his mobile telephone,
okay.
But it'll be me who'd be presenting some of the slides. So
okay, Brian. Okay, Brian, please go ahead. But I must remind you, you have 10 minutes for the presentation and 5 minutes for the questions and answers. Thank you.
Okay, thanks very much. Yes, okay, thanks very much, Francis. As I say, unfortunately, Godfrey is traveling between facilities at the moment, so he kindly asked me to present some of these slides. So I won't be as knowledgeable as Godfrey, but hopefully I can make up for that. So anyway, so really the background of this was obviously TB is a growing health problem, and we particularly see that in sub-Saharan Africa going forward. I think not necessarily for this presentation, but I think when we come later this morning to discuss what next for the MURIA group, I think one of the impact of COVID-19 will be on what's happening with non-infectious diseases like TB, malaria, etc. So maybe we'll come back to that. Anyway, we felt there was a need to determine the prevalence of drug-induced hepatotoxicity amongst these treatments to help to provide sort of future direction going forward, particularly because adherence to anti-TB treatments is crucial really in obtaining good outcomes. So the objective really was to ascertain the prevalence of anti-tuberculosis treatment induced hepatotoxicity and potential factors really associated with that in patients in Botswana. So we started this work in Princess Marina Hospital, which is a tertiary level hospital in Gaborone and probably the largest hospital and largest academic hospital in Botswana. So certainly will give us a good start to really start to assess the situation and to plan for future treatment. So what we did in one of the largest wards was really a retrospective study, cross-sectional study between 2017 and 2018. And anti-TB hepatotoxicity was obviously categorized using well-known scales, including the Rusev-Klofas Kuslav causality assessment method, RUCAM, etc. And severity was categorized according to the WHO criteria. And then we looked at any associations using binary logistic regression. So what will we find? We actually ascertained 128 patients, but in the end only able to determine or to collect data from 112 patient files. Of those, 13.4% had developed hepatotoxicity after an average of 20.4 days of treatment. And that's actually pretty similar to most of the published studies showing this sort of range of hepatotoxicity with the use of antimalarial treatments. So grade 3 and 4 hepatotoxicity was found in 2/3 of these cases. So when we did find hepatotoxicity, it was usually reasonably severe. So 86.7% would categorize having as possible ATT-associated hepatotoxicity. Patients with elevated baseline alanine transaminase were more likely to develop hepatotoxicity, and we see that in other studies. And interestingly, patients with normal hemoglobin were also more likely to develop hepatotoxicity. So some interesting findings there. And this slide here, so I should have that on full. I know it's slightly busy, but what we tried to do here was to look at a number with hepatotoxicity, number without hepatotoxicity, and look at the factors including ALT, AST, ALP, as you can see down on the left-hand side, albumin, white blood cell, hemoglobin, etc. So from these, we're able obviously to look at issues such as hemoglobin down at the bottom to see if there's a difference between the two populations, and right at the top, ALT between the two populations. So they were the two areas that we saw were significant. AST, ALP, etc. were not significant between the two groups in terms of their values. The main ones, as you can see, were ALT and hemoglobin, not platelet counts, etc. So really, I think what we can say there is normal hemoglobin and elevated baseline ALT were significantly associated with anti-TB hepatotoxicity, but not the others. So really, this was more or less a pilot retrospective study. So we'll be looking at some of these in more detail, particularly issues around hemoglobin and things like that. So that for us is a point going forward with this study. Just to say that we've now published the findings. So this was sort of, we got acceptance about a few days ago. And so now the Word document is now available and referenced in PubMed for anyone who's interested. And obviously we can send you copies as well. So that's all I've got to say. Any questions?
Thank you, Brian. Questions are invited, ladies and gentlemen. Evans Kajiso. Evans Kajiso, please ask your question.
Hello, can you
hear me? Yes, please go ahead, Evans.
Thank you. Will you be able to send us all the presentations?
Yes, this is Brian here. What we'll be doing, as we normally do with most of our MURIA conferences, is to make the presentations and the abstract booklet available online. Okay. So that's the idea, is when we finish, then the idea is to say, is we normally do, is make everything available online through ILSA and to group at Nelson Mandela. So we've already sent the consolidated abstract booklet, so hopefully that will go on soon. I think, as I say, of all 46 abstracts, and the idea is that we will make the presentations available as well. So we'll try and do that as quickly as possible.
Thank you so much, Gordon. I appreciate it.
Brian, on that note, Brian, would it also be possible for us to share the recording for the conference?
Yes, I think that should be okay. I'm not technically minded, so I don't know how we can do that, but yes, hopefully yes, we should be able to put that onto the website so people can hear as well.
Okay, thanks.
Thank you. Any other question, ladies and gentlemen?
Hi, hello everyone. My name is Dami Anosaya. I'm calling from University of Maryland, Baltimore in the US. Thanks for a great conversation. I came in a little late, so I don't know if this topic has been addressed, but I am concerned about data sources for medicines utilization research in Africa as a whole, maybe even by country. Do we know, have we been able to identify any longitudinal databases of patient level medication use that are accessible for research? Just wanted any comments from the group on that. Thank you.
Francis, shall I take that first?
Yeah, yes, please go ahead, Brian.
Okay, thank you, Dami, for that. That's a very interesting comment you make. I think there's a myth generally that high-income countries have good patient-level data. I actually work across Europe, South America, Asia, Africa, etc. And I can say that there are actually very few countries with good patient-level data. The best is probably the Nordic countries. Pardon? The best is probably the Nordic countries with Sweden, but across most of Europe, it's actually very difficult to get hold of good quality patient level data. In South America, it's probably only Brazil, but generally this data is not available to researchers unless they work closely with health authority people in these countries. As far as Africa is concerned, probably most of the data is basically only accessible going back and looking through patient records. The intention obviously going forward is to get good patient level data, but we're probably in the infancy in Africa as we are across most countries. And as I say, that's the intention going forward to build on what we're doing here. Probably in Africa in the public system, probably Namibia have some of the best data, but it's generally limited in most of the public healthcare settings. Probably with the exception of HIV, we've probably got better data on HIV than we have in some of the other areas, but generally it's not available unless you're working with the doctors in the clinics. I don't know if Joseph and Olianka, you want to build on that?
Yeah, Brian. I think you're quite correct. I think our last meeting in South Africa with ISPI, we dwelt a lot on the issues of databases, data sources for pharmacoepi research in Africa. And of course, we know that majority of the privately owned health insurance companies in Africa, especially in South Africa and other places, they have a lot of data, mainly secondary, that people use for research. But for public sector hospitals in Nigeria, for example, I know it's very difficult. You need to work extra hard to be able to get anything done. And the data from the public health programs, TB, HIV, now, of course, because there are programs that are funded, so you have a lot of data from that. But generally, it's difficult. So, Dami, I think that might answer some of your questions. But the essence is to work with big companies, health insurance, HMOs in Nigeria to be able to get some data, at least, that we can work on and make policies upon. Thank you.
A quick follow-up on that. Thank you, Dr. Fadere. And thanks for the comments too, Brian. Are there any examples, and this is probably to Joseph, are there any examples of research that has been conducted with HMO data? And like, do we even have one example or a case where HMOs volunteered or, you know, sold data for any research? Like, is there any precedent?
Yeah, so maybe I will leave that to Dr. Ogunleye to answer, because we've actually approached a couple of HMOs in the past 2 years to look at the possibility of working with them. And of course, we hit a brick wall, sort of, because you know, everybody is trying to protect something. They didn't really understand the importance of what we're trying to do. So Olayinka, are you there? Can you add to that?
I mean, you're very correct. You know, we've tried to reach out to them, and just like you said, some elements of suspicion here and there. But I think we're likely going to succeed, because what we did eventually is to get a buy-in of, you know, the registrar of, you know, their consortium. They have an association, okay, who perhaps, you know, seems to understand better, okay, and has some, you know, research orientation. But I believe, you know, if we progress with that, we might be able to. And again, the kind of data they also have, it's not as robust as one would also, you know, desire, but it could just be a starting point.
Thank you, ladies and gentlemen. I'm going to have to arrest the meeting at this point. And for further discussions in this line, which is quite interesting, I think can be done at a time when the meeting is closing. And I do hope Dr. Dami Onasanya will be available at the time.
Yes, please.
Yeah, I would like to hand over to Dr. Olayinka Ogunleye for the next presenter. Thank you.
Thank you very much, Francis. I think Kevin is returning to present next. I don't know whether, you know, Mr. Falamu and his team are ready. Maybe after this slot, we can have them. But next, we're going to have Mr. Kevin Awere returning to present to us comparative prevalence of hepatotoxicity and hyperglycemia of DTG and NVP-based regimen in HIV patients seen at Kenyatta National Hospital. Mr. Kevin, you're welcome back. Please, you have the floor.
Thank you very much.
You can share your screen and commence your presentation. Thank you. Mr. Kevin, are you there?
Yes, I'm here. I'm experiencing challenges in sharing the screen.
You just look for the green icon, share screen. At the bottom part of the screen.
Okay, thank you very much. So this was part of the previous study that I just discussed.
All right, thank you.
Only that this time we were looking at two aspects, hepatotoxicity and hyperglycemia. Again, the outline, have a brief introduction, look at the objectives, the design and method, the results, the discussion, and finally, the conclusion. I think much of this I've already said. Dolutegravir in Kenya started being used in the year 2017. and it came highly recommended because of the fewer side effects that are associated with it. And that led to a transition of patients from all the non-nucleoside reverse transcriptase inhibitors, nevirapine being inclusive, to DTG-based regimens. Now, our objective in this case was to compare the safety with regard to hepatotoxicity and hyperglycemia of the two regimens, the DTG-based regimen and the nevirapine-based regimen at the Kenyatta National Hospital. Now, this one was a cross-sectional analytic study. We still had two groups, one group on dolutegravir and another on nevirapine, with a common backbone ART of tenofovir disoproxil, fumarate, and lamivudine for ease of comparison. We obtained our data from both electronic and physical patient medical records. And in addition to this, to augment, we also carried out two lab tests. One of them is ALT, and the other one was random blood sugar. We also carried out patient interviews using a structured questionnaire. Our study population comprised of patients who had been on these two regimens from the period of June 2017 till April 2019. And those who were included must have been on that regimen for at least 6 months, must have had a good medication adherence to their medicine. And this is captured at every patient visit at the hospital. And they must also have given voluntary informed consent. Now, we excluded those patients who had documented liver disease or active hepatitis due to other causes, and those who had a history of liver disease before the start of their current regimen. We used purposive sampling for this study. The analysis we carried out was the chi-square test to test the difference in these two aspects across the two regimens. And we also did logistic regression analysis to measure the impact that other variables had on these two outcomes. Our primary outcomes were the prevalence of hepatotoxicity and hyperglycemia, and we graded this using the division of AIDS table for grading severity of adverse events, version 2 of November 2014. And that table has graded hyperglycemia and hepatotoxicity into mild, moderate, severe, and life-threatening, depending on the laboratory values, as I've shown. Again, we enrolled 111 participants, 86 on the DTG-based regimen and 26 on the NVP-based regimen. When we look at the baseline characteristics, for age, there was no difference between those two groups, but when it came to the sex and the duration of treatment, there was a statistically significant difference between the two groups. Now, when we looked at the prevalence of hyperglycemia, we did not find any statistical significance between these two groups with regard to that specific parameter. The dolutegravir arm of the study appeared to have a marginally higher prevalence of mild hyperglycemia of 5.3% compared to that of nevirapine-based regimen at 4.6%. And the moderate hyperglycemia, we found none among the participants in the nevirapine wing, but we found
minutes more. Thank you. 5 minutes more. Thank you.
Okay, but we found a prevalence of 1.3% for the participants who on the dolutegravir arm of the study. Coming to hepatotoxicity, we also did not find a statistically significant difference between the two regimens when it came to hepatotoxicity. However, we found a higher prevalence of mild hepatotoxicity among the participants in the nevirapine arm of the study, which was 16.7%, compared to that of the dolutegravir arm of the study, which was 10.8%. We do not find any cases of moderate hepatotoxicity on the nevirapine arm of the study, but we did find a 3.6% prevalence in the dolutegravir arm of the study. Now, like I said before, the study only found mild and moderate cases of these two aspects. We do not find any severe or potentially life-threatening hepatotoxicity or hyperglycemia. And again, there was no significant difference between the two parameters that we measured between those two regimens. I think I've just said that. Finally, our conclusion was that there was no difference in the prevalence of hepatotoxicity and hyperglycemia between the nevirapine-based regimen and the dolutegravir-based regimen. I'd like to acknowledge the study participants, the clinical and the laboratory staff at the KNH hospital, and the management, together with my co-authors, Professor Guantai and Dr. Oluka. Thank you, ladies and gentlemen.
Thank you very much, Mr. Kevin, for the presentation. We now welcome questions and comments, please. Questions and comments? Hello, anybody, questions and comments? Please, you can raise your hand so that we can recognize you. Okay, yes, Dr. Francis, please ask your question or make your comment.
Kevin, what was the duration, even though there was no difference between the two regimens in terms of the duration spent on the regimens, yes. What was the duration? I just want to see that, because when you talk about hyperglycemia, sometimes this is something we expect to come over a long period of time. And I think there has been some information regarding weight gain with dolutegravir, and therefore there is likely danger in the future for people taking dolutegravir probably experiencing diabetes. So I just want to know what you conclude in that particular regard, because the duration might be quite short for us to conclude the safety of these drugs is equivalent.
Thank you very much, Francis.
Mr. Carvey. All right.
As you rightly put it, for the dolutegravir.
You want to answer that?
Yes. For the dolutegravir arm of the study, the participants averagely had been on that regimen for between 1 to 2 years. And for the nevirapine arm of the study, they had been on that regimen for between 1 to more than 6 years. So as you rightly put it, for the dolutegravir regimen, a majority of them had not been on the regimen for longer than 2 years. And that could have had some bearing on our findings.
Okay. Thank you.
Thank you very much. Do we have further comment, question? Okay, let me just look through again if there's any hand that is raised. Okay, Ayub Hussein, please can you ask your question or make your comment, please? Hussein Ayub. I hope we're not facing the difficulty we had yesterday now. Mr. Kennedy Wendo, please, can you speak? Can you unmute yourself and speak? Mr. Wendo, can you unmute yourself and speak? Yes. Mr. Kennedy Wendo, please speak. Unmute yourself, please, and speak. Yes, you can speak. You are unmuted. I think, okay. Mr. Kennedy Wendo.
Yes.
And Mr. Hussein Ayub.
Hello. This is Wendo.
Okay, maybe you can type your questions in the chat and then I'll read them out. Can you type your questions in the chat and then I'll read them out if you are having difficulties with your audio. Mr. Hussein, are you there? Can you unmute yourself and speak if you're enabled? I think we're having difficulties with the participants being able to speak. So if you can type your question or type your comments, then I'm going to speak to them. I'm going to read them out. We still have just about a minute or two for this slot.
I think there are already a few questions on the chat that you can share.
No, the questions I can see were the ones that we have taken care of at the last presentation.
Okay.
There's no new question. Okay, maybe I will encourage those who have questions and comments to reach out to Mr. Kevin after this session. So we're going to move on to the next presenter in order not to waste our time. So I hand over to Dr. Francis.
I think, Dr. Ogunle, you should, because it is the same gentleman presenting.
Okay, so I just usher him in again.
Yes, please.
So please, Mr. Kevin, be on the standby and let me announce the next topic. So dear colleagues, Mr. Kevin would continue with a presentation on comparative effectiveness of nevirapine and dolutegravir-based regimens in HIV patients in Kenya, findings and implications. Mr. Kevin. Can we all unmute ourselves because we're having some background disturbances? So, Mr. Kevin, you're welcome on the floor again. Thank you.
Thank you very much. Now, this is also building on the previous two studies, but in this particular one, we focused on the effectiveness of the two regimens. And as I said earlier, dolutegravir is relatively new in Kenya. It's an integrase transfer inhibitor that blocks viral DNA integration. And when it comes to effectiveness, Major advantages associated with dolutegravir are a fast viral suppression induction rate. It is less susceptible to viral mutation. Therefore, failure rate is significantly lower. However, the effectiveness data is not as well documented in Kenya as in other countries. In spite of that fact, there is a scale-up of dolutegravir in our setup in Kenya. Now, our main objective was to compare the effectiveness in viral suppression of a dolutegravir and an nevirapine-based regimen. This was an analytic historical cohort study with two groups, one of them on dolutegravir and the other one on nevirapine-based regimen, with a common backbone ART of tenofovir disoproxil fumarate with lamivudine. Now, our assessment on effectiveness was based on viral load test results, which are routinely done at the Kenyatta National Hospital. And the main source of our data was from physical and electronic patient medical records. Our study population included patients who were on these two regimens, that is the DTG-based regimen and the nevirapine-based regimen, between the period of June 2017 to April 2022. And we included only those who had been on these two regimens for at least 6 months, and those who had a good medication adherence record for the same period and gave voluntary informed consent. And we excluded those who had missing viral load information. Our sampling techniques for this particular study was purposive, and our analysis, we carried out the chi-square test. We carried out the chi-square test to find if there are any differences in the effectiveness in viral suppression between these two regimens and logistic regression. 111 participants met the eligibility criteria and were included in this study. 86 of them were on DTG, 25 on MVP. They differed in terms of distribution according to the sex of the participants and the duration of treatment with their current regimen, but there was no difference when it came to the distribution in terms of age between those two regimens. Now, in our analysis, we divided our results into two. The first one being those participants who achieved a viral load that was below 50 copies per milliliter, and those who were below 1,000 copies per milliliter. And in both analyses, the dolutegravir arm of the study appeared to have a better viral suppression than the nevirapine arm of the study, with about 93% of those participants on the dolutegravir arm of the study achieving viral load below 50 copies per ml, and only about 56% achieving viral load suppression in the nevirapine arm of the study. For those who achieve viral load below 1,000 copies per ml, all participants who were on the dolutegravir arm of the study managed to do this, with about only 80% on the nevirapine arm of the study achieving the same. Now, we carried out a logistic regression analysis for a number of variables. And as you can see, the regimen, the current regimen that the participants were on had a very significant association with the viral load of the participants. And from these findings, you can see that participants who were on the dolutegravir arm of the study were 10 times as likely as those who were on the nevirapine arm of the study to be virally suppressed. Now, these findings mirror that has been documented in other studies, specifically the single and the spring one study. Though there has never been a head-to-head comparison of dolutegravir and nevirapine in terms of viral suppression, we did find studies that compared another drug in the same class with nevirapine, that is efavirenz, with dolutegravir. And you can see in the single study, dolutegravir-based regimen was still superior in terms of viral load suppression with a rate of 88% compared to that of the efavirenz arm. A similar result is replicated in the spring study, which compared dolutegravir and efavirenz with a backbone of either abacavir with lamivudine or tenofovir disoproxil fumarate with emtricitabine. And in that particular study, the dolutegravir arm of the study had a suppression rate of 88% versus that of the efavirenz arm of the study, which was about 72%. Now, when we interrogated our results, among the reasons that could have led to the lower viral suppression rates for nevirapine arm of the study is that nevirapine for a long time was used as a single agent for the prevention of mother-to-child transmission. Again, it has been used for a longer duration of time, and it has been documented that the NNRTIs, and specifically nevirapine, have a lower genetic barrier to mutation, and therefore resistance occurs easily. Our study was not without limitations, one major one being a smaller than projected sample size that only affected the nevirapine arm of the study. We therefore concluded that the dolutegravir-based regimen was superior in viral load suppression than the nevirapine-based regimen, and therefore this study supported the decision of the government of Kenya to transition all patients from nevirapine-based regimen to dolutegravir-based regimen. As I close, once again, I'd like to acknowledge the study participants, the clinical, the laboratory staff, and the KNH management in general, and my co-authors, Professor Godman, Professor Guantai, and Dr. Oluka. Thank you.
Thank you very much once again, Mr. Kirby, for this very stimulating presentation. So we'd like to welcome comments and questions on this presentation, please. Okay, so I have Mr. Hussein Ayub, Dami Onosoya, and Samuel Sine. So let me call Hussein Ayub first. Please, you can unmute yourself and speak, please. Hussein Ayub. Can you please unmute yourself and speak? I think colleagues are still having difficulties with speaking.
Yes, I'm a little bit. Yeah, I got a little bit busy. Can I call it back?
Mr. Hussein Ayub?
Yes, I'm little. Yeah, I'm a little bit busy now. Can I call it? Can I call it?
Hand. Do you want to ask a question?
Yeah, I wanted to ask a question, but I'm a little bit busy. Can I call it later, or write my question?
Okay, you can write down your question. We don't have all the time. You know, we're going to move on to another presentation. You know, soon. So can we Mr. Samuel Sine. Mr. Samuel Sine. What about Dami Onosoya? If you're there, please, can you unmute yourself and speak?
Okay, thank you, Dr. Kenneth. I'm sorry I missed the last name. I think Dr. Wenel. That was a great presentation. I had a question about the study design in terms of inclusion criteria. So you mentioned that all participants needed to have been on treatment for 6 months. Which treatment was that? Was that the TCG that everyone was taking, or dolutegravir and nevirapine? Where I'm really getting to is, was it a new user design where the participants in the study all starting dolutegravir and nevirapine for the first time in your study?
Okay, Mr. Kevin, can you take that?
Yes. The 6 months, what I meant with the 6 months is that the participants must have been on either of the regimens. That is, for those who are on the dolutegravir arm of the study, they had to have been on that regimen for at least 6 months. And those who are on the nevirapine arm of the study, the same thing. They had to be on that regimen for a minimum of 6 months.
I see. So thank you for that. So I think the results are interesting, but as you mentioned in your limitations, dolutegravir is a newer drug, and so it could be possible that prescribers were prescribing it to those who were newly diagnosed with HIV. And if those who are newly diagnosed have higher viral loads at baseline compared to those who have been taking nevirapine for all the years that nevirapine has been in the market, we could be actually observing higher viral loads in the nevirapine group that are unrelated to dolutegravir treatment, and lower, and in the same vein, lower viral loads in the nevirapine group that are not that are just because they've been on treatment for a long time, or they've been diagnosed with HIV for a long time. And I do see that you mentioned that in your discussion. So I'm a bit wary about the 10 points relative risk reported, in view of this time lag bias, I think.
Mr. Kevin, last comments?
Yes, as he has rightly put it, there was a significant difference in the duration of time that participants on the two regimens had been on their regimen. But that's notwithstanding, those on the dolutegravir regimen, despite having been on it shorter, still observed a higher viral load suppression rate in that arm as compared to those in the nevirapine arm of the study who have been on their regimen for a longer time. And I think this just goes further to show the superiority of the dolutegravir regimen in viral load suppression.
Mr. Kevin, there's a last question here we're going to take before we move on from Bernard Owino. He says, what's the reason for the small sample size of the nevirapine arm? Yet during the period 2017 to 2019, the regimen was still commonly in use.
Thank you for that. The reason for the small sample size in the nevirapine arm of the study was occasioned by a policy shift by the government of Kenya, the Ministry of Health, that accelerated the transition of patients from the nevirapine-based regimen to a dolutegravir-based regimen. And therefore, what this meant was that by the time we were collecting the data, a majority of them had moved from nevirapine to the dolutegravir regimen, and therefore they did not qualify. I hope that answers your question.
All right, thank you very much for that. We're going to revert to the earlier presentation that was skipped. And I'm going to invite Dr. Francis to kindly introduce the topic. They're ready now. Dr. Calimera?
Yes, please.
Mr.
Yes,
Folaman and his group, they are ready now.
Okay, great. Ladies and gentlemen, I would like to introduce Mr. Nokwanda Vilakati and Mr. Fola Amu, who will now take us.
She's interestingly a woman.
Oh, okay. I do apologize for that. I'm looking at the program and it says Mr. Okay, but I apologize. And I'd like to invite them to present physicochemical assessment of selected brands of artemethyl-lumefantrine tablets obtained from retail pharmacies in Mbabane, Eswatini. The floor is yours. You have 10 minutes for the presentation, and we'll go through a 5-minute session of questions and answers. Thank you very much.
Thank you very much. Greetings to you all. I'm Nokwanda Vilagati, and apologies for the technical issues. I'm here with my supervisor, Mr. A. Amo, on the presentation of physicochemical assessment of selected brands of AL tablets obtained from retail pharmacies in Mbabane, Eswatini.
You please make it a full screen. Thank you.
Yes, this is the outline of what I'll be presenting on. So physicochemical assessment involves all the physical and chemical activities that are required to ensure that the medicine reaching the patient is effective, safe, and also of acceptable quality. In accordance with WHO recommendations, the first-line treatment for uncomplicated malaria is AL, which was selected for its tolerability, low adverse effects, and also less resistance. In terms of drug category, antimalarials and antibiotics are said to be among the most widely reported counterfeits. This is supported by the study that was done in Angola, whereby 33 million of AL tablets were found to be counterfeit, and those were enough drugs to treat more than half of Angola's yearly malaria cases. Problem statement. So there have been a lot of death cases. There have been a lot of death cases due to the use of counterfeit or substandard antimalarials, which is supported by the study done by WHO in sub-Saharan Africa. And the Eswatini Medicines Regulatory Authority has been developed, but is not yet fully operational, which then causes a potential danger to the use of those counterfeit antimalarials. The rationale of the study. Eswatini is one of the African countries chosen by the World Health Organization to eliminate malaria by the year 2025. Various interventions have been undertaken by the country in its quest to eliminate malaria, but even so, the malaria cases are still rising. And in Eswatini, there are no scientific published documents on the quality of AL tablets, which therefore makes the study vital in the provision of that information. Counterfeit and substandard antimalarials can cause a lot of problems for the economy and also for the patients, including the problems of economic sabotage, therapeutic failure, lack of trust and confidence in the healthcare system, and also serious side effects or death. The significance of the study. Findings from the study might help ensure the safe and effective use of good quality AL tablets that are imported into the country. And the Ministry of Health might aid drug assessment studies as one of their interventions in their goal to eradicate malaria. The purpose of the study was to assess physicochemical properties of selected brands of AL tablets obtained from retail pharmacies in Babane. And the objective was to perform this physicochemical test on the selected brands in order to determine if the selected brands are counterfeit or not. Setting. This study was carried out at EMCU using the University Dry Laboratory. For the population, only brands of 20/120 AL tablets were used, and the study used non-probability sampling. Those are the 5 tests, physicochemical tests that were carried out on the tablets. And these are all the instruments that were used during the tablet assessment. For analysis, Microsoft Excel 2013 was used. Anonymity was ensured by using codes for naming the selected brands. For validity, SOPs were followed and a pretest was done before each test was carried out. So after the test, results were recorded and analyzed. Table 1 shows uniformity of weight, which is the assessment of how ingredients vary in tablets or capsules. When looking at the table, we see the mean weight of 20 tablets that were weighed individually. The BP specifies that the individual weight of tablets should not deviate from the average weight by plus or minus 7.5. And looking at the standard deviation, we see that both brand AL01 and AL02 passed the uniformity of weight test. Table 2 shows hardness test.
You have 5 minutes.
Hardness being
5
hardness
minutes.
is the load required. Hardness is the load required to crush a tablet to determine its breaking point and its structural integrity. Sorry. When looking at the table, we see the mean hardness of 10 tablets that were assessed individually in newtons and also in kilograms. We see that brand AL01 passed the test because it is within the specified limits of 5 to 8 kgs per British Pharmacopoeia, whereas brand AL02 failed the test because it is below the specified limit. And this may be because of the amount of binder that was used, the compressive force of tablets, and the method of granulation that was used during manufacture. Table 3 shows friability test, which is used to assess the tablet's ability to withstand the tendency to break, crumble, or fail to maintain its integrity after compression. Looking at the table, we see the friability percentage loss, which is less than 1%, meaning that both brands pass the test as they are within the specified limits for friability. Table 4 shows disintegration time, which was done 3 times. Disintegration being the time required for a tablet to break into particles under a given set of conditions. And looking at the table, we see the mean disintegration time is below 15 minutes, which means that both brand AL01 and AL02 passed the test. Graph 1 shows dissolution rate. Dissolution is a standardized method for measuring the rate of release from a dosage form. Here we have the tolerance limit for artemether, stating that not less than 45% of the labeled amount of artemether must dissolve in 60 minutes, and for lumefantrine, not less than 60% of the amount must dissolve in 45 minutes. And looking at this graph, we see that brand AL01 passed the test for both its artemether and lumefantrine content because in 60 minutes, more than 45% of artemether was dissolved, and in 45 minutes, more than 60% of lumefantrine was dissolved. Moving to brain AL02, we see that this brain failed the test because at 60 minutes, less than 45% of artemether was dissolved, and at 45 minutes, less than 60% of lumefantrine was dissolved. And this might have been because of the amount of lubricants that were used, the disintegrates used, and also the size of solid particles in the solution. In conclusion, out of the two brains, none was found to be a counterfeit, since brain AL01 complied with all the pharmacopoeial standards. However, brain AL02 was found to be substandard because it failed the hardness and also dissolution rate, which suggests that the brain AL02 circulating in Eswatini is of poor quality when looking at only the physical-chemical properties. And this then presented danger to the health of the Eswatini people. The researcher would then recommend for the government of Eswatini through the Ministry of Health to enforce an operational and vigilant medicines regulatory authority, so to totally eradicate counterfeit and also substandard drugs. Thank you.
For a wonderful and I believe a stimulating presentation. I'd like to invite questions. I'm seeing hands. I'll ask Dami Onansanya to ask the question, please.
Oh no, I apologize. I didn't lower my hand from the last question.
Okay. Oh, okay, great. David Sampson, please. You may ask your question. David, will you please unmute and ask your question? David Sampson? Okay, I'll ask.
Francis, can I just make a comment, a quick question while David is?
Okay.
Yes, so thank you very much for this very good study. I'm just wondering, is physical chemical assessment enough to judge the genuineness or otherwise of a formulation without doing a bioequivalent study to complement the physical chemical properties? Just drawing from, the statement you made in the conclusion. Will it have been better to do complement this study with a bioequivalent study, because there are some questions that are not answered beyond the physical chemical properties?
Thank you, Dr. Olainka. I had the exact same question, asking whether because she judged the drugs are not, they're both not counterfeit. And I was wondering what they find out. Does it really influence the clinical outcomes when they are used? I think it is a similar question to yours.
Okay,
so please kindly respond.
Thank you for your questions. So the drugs were found to be substandard when only looking at the physical-chemical properties, but the researcher would recommend for more studies to be done, more in vitro and also in vivo studies, so to determine the quality of the AL tablets.
Thank you, colleagues. Just to add on to what has been said, within the scope of time we had, we could only focus on the physical-chemical characterization. And based on that, we were able to draw that conclusion. But from what has been said, we have noted it. We also appreciate the fact that we need to do further studies. But it was just a project for undergraduate program. So we had to, time became a limitation. So we could only cover the physical-chemical characterization at that point.
Okay, thank you very much. Any other?
Comment is predicated on the conclusion that is sounding emphatic that it is not counterfeit. It's a good study and it's well received, but we can't conclude, you know, that it's not, the products are not counterfeit if you have not really go through the old orb.
Okay.
Over,
We
Francis.
appreciate that.
Okay.
Thank you.
Thank you, Dr. Olainka. Isaac Lusenaka, please ask your question. Unmute, please, and ask your question. Isaac is off.
Sorry, I didn't have a question at the moment.
Oh, okay. Thank you. Okay. In the interest of time, and I'm not seeing any hands raised, I'd like to hand over to Dr. Olainka to present the next speaker.
Thank you.
You're welcome.
Okay, thank you very much, Dr. Francis. And coincidentally, the next speaker is no other than Dr. Francis Kalimera himself, who is going to be discussing Tenofovir fumarate-associated nephrotoxicity, a retrospective cohort study at 2 referral hospitals in Namibia. Dr. Francis, you have the floor, and I'm sure you are well acquainted with the rules. Thank you.
Yes, I am. I'd like to share my screen. Okay, thank you for this opportunity. I present on Tenofovir disoproxil fumarate associated nephrotoxicity, a retrospective study at 2 referral hospitals in Namibia. I'd like to thank my co-authors, colleagues at work, my family, and of course, my creator. What I'm presenting is a paper that has been published with pharmacoepidemiology and drug safety. For more information, you may access the paper there. In the pre-ART era, there was a high incidence of HIV-associated renal disease, but this reduced because of ART. And Tenofovir, the drug that we are using a lot in our first line, is associated with nephrotoxicity. This drug is secreted in the proximal tubules, where it is believed it causes mitochondrial DNA polymerase gamma, falling within the nucleoside reverse transcriptase inhibitors. And the incidence of TDF-associated renal impairment is set at around 1.8 to 16.2 per 100 patient years. And there's varying evidence on the factors associated with those toxicities. So the objectives of this study was to estimate the incidence rate of decline in eGFR and the incidence rate of new onset renal impairment, and to identify the risk factors. We included HIV positive patients who were receiving TDF containing ART, were 16 years and older, with renal function test results available. And the data was gotten from the electronic patient management system. We defined a decline as a 25% drop in eGFR or greater than or equal to 1.5 increase in serum creatinine, in addition to transition to a lower eGFR stage. And then new onset renal impairment was defined as an EGFR less than 50 mils. We described the continuous variables with means and standard deviation. For the categorical variables, we used Pearson chi-square and Pearson and Spearman correlations to look at the relationship between the variables. We calculated the incidence rate using the OpenAP calculator, and to identify the risk factors and predictors of these adverse events, we used Cox regression analysis with backward stepwise method, and we carried out sensitivity using binary logistic regression analysis because the time variable was rather unstable. Confidence limit was set at 95% and significance at a p-value less than 0.05, and the study was approved. We included 10,387 patients. And the majority of these patients at baseline were in stage 1 and stage 2 of EGFR. The baseline EGFR was 72. The males had a higher baseline EGFR. The age of the cohort was 39. The males were older. And for the variables that we considered during the follow-up, we had more males switch to second line, that is with the PI regimen. More males had TB, had to take TB therapy. More males had a less than 200 CD4 count. More males had a viral load greater than 1,000. And also more males had hyperfiltration. The majority of patients who experienced decline were in stage 1 and stage 2 at baseline. And then we have an increased number of patients at follow-up in the stages 3, 4, and 5. And then pregnancy and hyperfiltration were related to the decline. Considering the renal impairment, we had more females experiencing renal impairment, and then more patients who were in stage 2 than in stage 1 experienced renal impairment. And then also the pregnant females were more associated with that. And then the decline was, we estimated it at 4.5 cases per 100 patient years. But when we unpacked it to the different stages of EGFR, we saw that the decline occurred much more in patients who were in stage 1 at 9.9, then stage 2, 4.6, stage 3, 2.3 per 100 patient years, while stage 4, we have 8.2 per 1,000 patient years. So we compared these incidence rates, and we found that for the new onset EGFR needing dosage adjustment, we had more experiencing this decline in stage 2 compared with stage 1 being used as a reference. Again, for those with new EGFR in stage 3, more in those who are in stage 2 at baseline than in stage 1. Again, more in stage 4 for those who are in stage 2 than in stage 1. But then those who went into stage 5, there was no difference between stage 1 and stage 2. And then for those in stage, who went into stage 4 from stage 1, compared with those in stage 3, we had no difference, and there was no difference in those going into stage 5. The predictors were generally the first 5 as you see on the list, but the greatest magnitude of effect, the effect size with EGFR baseline greater than 60 was quite large, 15.6, as shown with the hazard ratio, followed by hyperfiltration and then by pregnancy. And then the predictors of renal impairment, we have hyperfiltration and then followed by pregnancy as with the greatest effect sizes. And the Kaplan-Meier curve shows you that stage 1 was at greater risk, followed by stage 2 of the decline. So the high baseline, EGFR being a risk factor for decline, is not a new finding. It has been found with other studies. Some of them say the patients start ART with a low body weight and therefore a low serum creatinine, and there is a a view that these patients had a proper renal function at the beginning, but as ART starts, it gets worse. But then our finding, the patients really had generally equal, no difference in body weight. So we put it at the finding that patients must have had a higher expression of organic anion transporters in the basement membrane of the proximal tubules if they had a high baseline EGFR, therefore allowing higher concentrations of tenofovir in the proximal tubular cells and leading to damage of the mitochondrial DNA polymerase gamma, resulting in a reduction in EGFR. Then for hyperfiltration, this is a known association finding for patients who are pre-diabetes and pre-hypertension having hyperfiltration, and when they're exposed to a nephrotoxic drug like tenofovir, you would expect a decline in EGFR. And then for female gender, they do experience, because of HIV, premature ovarian insufficiency, resulting in hypoestrogenemia. And this reduction in estrogen is associated with a decline in renal function because estrogen is a known renal protective steroid. And therefore, because of HIV, the females are more prone to EGFR decline. Now, pregnancy and weight, pregnancy especially was the one difficult because we have increased concentrations of estrogen in these patients, but because of pregnancy, then pregnancy was a risk factor for the decline and also for renal impairment, and this could be associated with autocoids that do increase in pregnancy. And then weight also associated with diabetes and hypertension could be a risk factor. A high baseline EGFR is a predictor of decline in EGFR, but is protective against new onset renal impairment. Our recommendations, in addition, or affirming recommendations that have already been made, is to use alternatives to TDF in patients with hyperfiltration. But if that is not done, prompt monitoring of renal function is advised because these patients, as you have seen from this study, the decline is quite severe and get into stages of EGFR that require dosage adjustment. So instead of using the usual testing saying after 6 months or after 1 year, perhaps these patients with hyperfiltration, the monitoring should be sooner. Then hyperfiltration and high body weights with low baseline EGFRs should be targets for diabetes mellitus and hypertension assessment because we found with the correlation analysis high body weights with low baseline EGFRs. And then lastly, prompt observation of increases in EGFR and increase of those in TDF in patients with improved renal function is advised because patients' renal function improved from lower stages to higher stages that require dosage adjustment. Thank you very much.
Thank you very much, Dr. Francis, for that wonderful presentation. I'd like to welcome comments and questions from colleagues. Comments and questions from colleagues, please. Please, can I see your hand of the comments or question to ask so I can recognize you? Any comment or question, please? Okay. Let me see if there's any typed in.
Probably people are wanting their break before the last session.
I see Felicia Williams.
Yeah.
No comments or questions?
I see Felicia Williams who would like to ask a question.
Okay.
Hello, can you hear me? Good afternoon.
Felicia, please, can you speak? Yes, we can hear you. Please speak. You're welcome.
Good morning, good afternoon, good evening, wherever you are in the world.
Felicia, please speak.
Can you hear me?
We can hear you. We can hear you, Felicia.
I said good morning, good afternoon, good evening, wherever you are in the world. We appreciate the presentation.
Oh, good night too. Thank you.
Oh, good night, all of the
Yes, we
above.
can hear you.
We really appreciate the presentation. The question I wanted to ask has to do with, did they consider confounding variables that could affect the outcome of the research done?
Okay.
Yeah, that's my question.
Okay, thank you. Well, the issue regarding the confounding variables, because the study was based on patients taking TDF, and first of all, we could not include other regimens because we used Ministry of Health data. And the Ministry of Health data is patients who have renal function results are those who are taking TDF. They wouldn't do tests on patients who are not taking TDF. So we could not include patients who have patients who are taking other regimens. So that could be maybe a confounding. But then one thing for sure is that carrying out post-Cox regression analysis and binary logistic regression analysis does help to cater for possible confounding. The other thing is that the patients having hyperfiltration and which are probable, which is known to be a clinical event that does come before patients are diagnosed with diabetes and with hypertension, could be protopathic bias, so that the patients were likely to get renal impairment anyway, and so they were at risk of getting nephrotoxicity with TDF. But the fact remains that we did carry out analysis of, we excluded patients who had hyperfiltration and carried out analysis in patients who had a normal renal function. And then we found, that that still TDF was a risk factor. And so including these patients did help just to increase to show the results as they did appear that hyperfiltration is a risk factor indeed. I hope I've answered the question.
Thank you very much, Francis. Any other comment or question?
Just quickly, thanks, Dr. Kalimera. That was a very interesting. That was a very interesting presentation and very good research there, with a nice data source actually also. Were you able to determine in the MOS data when people started the previous line treatment? So from your presentation, I think you may have mentioned that TDF only, or maybe I saw it in the article, that TDF became first line treatment in July, and people were taking zidovudine-based regimens before that time. So my question is, you know, even amongst TDF users, a TDF user is different from other TDF users if they have been taking zidovudine for a longer period compared to others. So are you concerned that some of the effects you are reading for TDF use may actually be carryover effects from the length of time some people in the study had used zidovudine compared to others?
Thank you for your question. The patients who are included were those who initiated TDF as their first ART, were ART naive at the time. So we did not include patients who were switched from other regimens to TDF.
I see. Oh, that assuages my concern.
Yeah, no, thank you.
Can I put a question, Francis?
I will agree, even though I am not chairing this. I'm not chairing this session.
Okay, please make it brief and quick.
Okay.
Ready.
Thank you for that very, very scientific presentation.
Okay.
Looking at it, the outcome has quite a bit of clinical implications in terms of patient management and also influence of policy. Have you taken any steps to institute change in the clinical practice with respect to TDF?
Thank you for the question. The paper has just been published, and I'm now going to present the results to the Ministry of Health and Social Services.
Thank you.
Yeah.
Hello. Hello.
Fantastic, Francis. Thank you very much for that highly stimulating study.
Hello. Can you hear me? Can you hear me? Hello.
Can I hear you?
Yeah.
Yes, I can hear you.
Yeah, I have a suggestion. He can include TDF naive patient as inclusion criterion to exclude those that are not naive. Did I communicate properly?
Yes. In the first place, this was a retrospective study. And then if we are to include TDF naive patients, those patients do not have renal function results. In Namibia, they only do pre-ART EGFR for patients who are going to start TDF containing regimens. That is where those patients were not included.
Okay.
Yeah.
Thank you.
You're welcome.
Francis, this is Brian here. I suggest we only got 5 minutes to the plenary session, so maybe we can break now and then come back to that. Or Francis and Joseph, etc.
Okay, thank you.
So see you all shortly soon. Got about 5 minutes for a quick comfort break and things like that.
Okay, thank you.
Brian, you may have to give my apologies for the next session.
Yeah.
We have an urgent meeting with the university management at 2:30 Kenyan time.
Okay, no problem. Anastasia, I've just seen your feedback form, so I'm just about made the odd comment to send to send to John, ready to send out. So we'll do that in as well shortly.
Thank you very much.
Brian, I would also like to register apologies because I have to leave on an urgent matter at the moment.
John? Okay. Hello? John? Nothing.
Yes, okay. Hello, hi Brian, can you hear me?
All right, okay, yes I can, yeah. So what I'll do is share one slide in a moment, but Joseph, do you want to start this session off? Hello, Joseph. answer. Yeah. Well, it's now sort of wherever you are in the world, it's sort of 12:30 UK time, 1:30, I guess, South African and Nigerian time. So firstly, thank you all very much for a very productive 2 days. So what we'll do in the last session, before we hand over to Dan to close the conference, will just be to go through, obviously, some of the key areas going forward that we can work with as a group, if you like. So that's what we'll do in the next sort of 45 minutes or so with Joseph and myself. So what I did here was just put up some slides to kick it off, and then please feel free through Joseph, through putting your hand up to then suggest sort of ways forward as we go through. So all I've done here is just to put up some slides, if you like, or a slide on what, between Joseph and I, we quickly thought of going forward to be a starter for 10. Firstly, to me, as I said this earlier today, that we've had over 300 participants today. So that's a record for us going forward. And I think it shows the value of having this type of virtual meeting where more people can participate than if we meet face to face. So I know that we'll be looking to distribute some certificates, and we've been working with John to look at some feedback forms. So John, what will be the logistics around that?
Once the forms are sorted out, we are going to generate a survey, a Google Doc, which we are going to share with the participants. Because we have the email address, they'll be able to complete that, and once they complete that, they'll also give us the details, how they want their names to appear in the certificate, and that's what we use for completing the certificate.
Okay, thanks for that. So that's an incentive, if you like, for everyone to feedback, and we certainly want your feedback. The reason being is that one of the outputs from the last 2 days will be, we'll look to submit a report to one of the journals. The first 2 meetings were submitted to Expert Review of Pharmacoeconomics and Outcomes Research, so there's no reason why we can't do that with the last 2 days, particularly given the wealth of presentations. I think we had in the end some 46 abstracts, which was wonderful given the relatively short time. So I think that just shows how active we are as a group. And over the last 2 days, we heard a lot about some of the new research or researchers ongoing and new researchers planned. So I think that's wonderful. So I just put down some themes here, I think, that we can discuss over the next sort of 40 minutes or so, and then we can go forward and plan what we're doing for the future. Firstly, apart from, I think in the questionnaire, we will ask what should be the future of the meetings, and I put down some things there so we can build on that. I think firstly, it would be useful, and we heard a lot about a number of studies ongoing, if we had some way of collecting or collating where you're looking for collaboration or help outside of your university. So if you're planning a 2-country study or greater, then it'd be good to see if we can try and collate some of those. We had some discussions about warfarin in I think yesterday late morning in one of the theme groups, and talking about quality indicators and things like that. So to me, that's a great opportunity going forward, is if we're planning studies, then who else would be useful to collaborate with? And that will enhance not only the visibility of your group, but will also enhance the chances of publication in a higher factor journal. Certainly, in the more cross collaboration that we have, and the more interested is that paper is to a international journal. And that will also help us in grant applications. We've obviously got several grant applications ongoing at the moment. So it would be good again to have a means of collating that. Maybe we come back to that. Newsletters, Ollie Anker's done a good job, so should we keep those going? What about future workshops? Maybe we can have sub-themes, so around how you develop, say, quality indicators, how you do more sophisticated drug utilization studies, how we can get access to or improve access to some of the databases and things like that. Along with, at the moment, we're progressing well with a handbook on drug utilization studies through Hanley, Hanata, and Ilse. So maybe that's another way of going forward. So I just put down some activities there and suggestions for the future, but probably now it's a good time to throw it open. So I'll stop sharing this screen. As I say, these are some of the ideas going forward, and then open it up to the floor. So Joseph, do you want to take over and start progressing discussions? And I can jot some things down ready to summarize towards the end.
Okay. Thank everybody for the past 2 days, and for those that we've communicated over this period, I apologize if I've sort of hurried you up or somehow. Well, actually, it was to make this thing happen. And we want to thank our team from Nairobi, Professor Guantai, and our team, John, for the ICT backup. Without this meeting wouldn't have been possible. And for everyone for taking part. But like Brian said, the essence of the 30 minutes talk or discussion is to chart a way forward. So Brian has brought forward a lot of ideas, and I will leave it open for now for people to contribute. And then we can, for the next 10, 15 minutes, for people to contribute. And then, so if you want to say something, you have an idea, could you just signify by raising your hands, and then unmute yourself, and then you talk. Brian, can you see any hands raised?
I can't do it on my screen, so maybe somebody just wants to shout out their name and go from there. So who would like to start us off on potential ways forward?
Okay. So while you write maybe your name and what you want to say on the chat, I think the first thing, one of the things we need to do is, we brought up a lot of ideas, a lot of studies from different parts of Africa and beyond. So I think there is a way, we need to find a way to group people together. People that are working on similar themes. There has to be a way of linking ourselves together so that we can collaborate. So collaboration is the first thing. So how do we do this? Do we need a unit, or do we need people to communicate with themselves? Of course, we are going to make available the emails of everybody, all the participants, and it will be good if they put their affiliations also, so that people can link up with one another even after this meeting. So that's very important, collaboration. So any comments?
Right. So I'm Kwame speaking from Ghana.
Okay.
Can you hear me?
Yes, loud and clear.
Right. Thank you very much. Yeah, I think it's been quite stimulating discussions, very, very interesting research works, some very interesting findings relevant to quality improvement of practice, policy, etc., etc. But I think some of the quality of the abstracts and some of the presentation were also not very good. We have to work on that one. I mean, we should be able to, lift up the quality of this so that, works that are presented here, the methodologies used, etc., is quite robust enough to generate us findings that we need, to impact on the health systems, in Africa. Now, on the issue of collaboration and joint grant applications, etc., I think it's the way to go. And also, that's also one of the ways of strengthening, some of these things so that we generate critical, evidence, which, we can use, whether to inform training, to inform policy, to inform practice, etc., etc. Thanks very much to the organizers,
Thank you, Kwame. And thank you for your very good presentation yesterday. And I think you guys in Ghana, you are a little bit way forward in the issue of antimicrobial stewardship and this. Yeah.
Okay, can I make
okay. Amand, okay. Is that Amand?
It's Amand here, Joseph.
Okay, okay, fine.
Yeah, in terms of, I think the grant application and doing kind of collaboration is really important, and now it's happening based on kind of the personal or proactive approach that some of us are using through Brian's kind of connection. But I think it'd be useful, if possible, to have some sort of a hub or a platform where people can, share their interest in doing a particular research and asking for people who are interested in collaborating in that particular research. And that will be the point of contact where people can make us teams and work on a particular grant. So I don't know if it's possible to use some of the university's hub, or I don't know, that people can share their research ideas and looking for partners across Africa. And then that would be the point where they can make a smaller team and apply for different grants.
Thank you, Amand. That's a very good idea. And I think now we're in the age of social media, and people communicate faster and easier with social media, even better than using emails. So again, looking at that, is a suggestion, could we have a kind of MURIA collaboration WhatsApp group? And anybody that needs information, that has information, so that could be a kind of collection point. People can send in what they are doing, who they want to collaborate with, and anybody in that network can link them up. Or instead of using emails directly, because most people don't open their mails for days, but most people look at WhatsApp every day. So we'll set up something like that, at least not just for general MURIA membership, but specifically for such collaboration. That's an idea and a suggestion. What do you think, Amanj?
Yeah, I think that's possible. But I mean, in terms of in terms of how people are willing to share their numbers as a group, personally, I don't mind, but not maybe everyone is comfortable with that. So that's absolutely an option. But I mean, ideally, I don't know if, for example, I don't know, MURIA currently is held on the Nelson Mandela's University, I think, website thing. So is it possible for them to create some sort of platform or thing like that provide that interaction without people providing any personal information like mobile numbers or things like that? I mean, the WhatsApp is absolutely an easy option to go for, to start with.
I don't know, is Ilse on the line at all? I know she was speaking this morning. No.
I can't see her.
So maybe that's what we can go back to, building what a man said.
Yeah.
I think we can probably do two. We can I think probably do two. One is we can do. Lynette, can you mute? We can probably do two. One is, if we can, to maybe one is as the man says, is to use, if we can get a hub to start that process going, and then we can do a WhatsApp group for people who are interested in particular grant applications. Hello, Lynette.
So, Brian.
Yes.
Ilse is there, but I think she's in the participant list, not the speakers list. So, I don't know how we invite her in so she can speak as well.
I think she needs to raise her hand, and then she will be notified to unmute herself, and she can speak.
Or just speak. I mean, there should be able.
No, I mean, Brian, you have because it's a webinar.
All right. So
participants cannot speak. You have to raise your hand, and the John from the KNH will give the permission to unmute and talk.
Yes, I'm doing that once I see them raising their hands.
Yes, or else I need to raise
check for
the raise
Ilse.
hand function.
Alex, I am here.
Okay.
Yeah, you are here now.
Oh, fantastic.
The question was, MURIA is still hosted on Nelson Mandela University website. That is fine. I've asked for the abstract compilation to be uploaded. So hopefully within the next hour or two, that will also be available. Just tell me exactly how you want me to, because I need to ask permission, obviously from the webmaster, but it is doable. I really think it is doable. So I will definitely support whatever is requested on the Nelson Mandela University website.
Aman, do you want to go forward then?
Yeah, I think Elsa was just kind of suggesting if it's possible to create some sort of a hub or a platform within the MURIA website, on the Nelson Mandela website, where people can propose ideas, looking for partners on a particular project. And then that will be the point of contact. Then people from there, they can make teams going forward.
I will come back to you. Just give me, it is today, Thursday, give me until Monday, Tuesday, and I will come back to the group with an answer, but I'm sure it is possible.
Okay, that should be great, I think. Thank you.
Yeah.
Thank you. Thank you very much. So that's about creating a group. I can see some comments also here from Kwame saying that we can use both platforms, just like Brand says, because it's important we communicate and we keep in touch. And that's very important. Any other person raising their hands for comments? Let me see.
I think maybe, Joseph, one other thing we could do, is then if possible to have some subgroups based on people's expertise and research interests. So for example, maybe you have a antimicrobial resistance and you have a adherence group. And then when it comes to the point of contact, if I'm doing a research in adherence, I will kind of contact that group, a bit more efficient. Just a thought.
This is Brian here. I think that's very good because we have seen different research going on in different countries. I was thinking quite a lot of research, obviously, as Aman says, around adherence, and we've heard presentations on pictograms. We've heard sort of different ongoing programs to address how we get patients access to medicines, particularly at this time. So I think that would be great because that will allow us, A, to cross-collaborate, and also B, if we got grant applications that are enhanced by having more than 2 or 3 countries, then we've got a ready group of people we can go to and saying, okay, this is your research area, what have you published on, and can you help us going forward to move that forward? So yes, and I think Amand, we've got a project on adherence at the moment. So do you want to talk about that?
I think that just is about the kind of with frontiers in pharmacology, which some of the peoples in MURIA are the co-editors, so Brian, Joseph, Lisa, also Maribel. And this basically just, I think, I would imagine some of you already received invitation to participate in that topic, just a way of disseminate your research around interventions to improve the management and adherence to medication for non-communicable diseases across Africa. So we can use that platform again to disseminate the findings from your research and also working out adherence and non-communicable diseases.
Okay, thank you. I can see Annelie's hand is up. Annelie,
Hi.
John, yeah.
Thank you, Joseph. You know, one thing I'm thinking about with COVID-19 now and the COVID-19.
Hello, Annelie, you're on mute. Can't hear you.
Good. Okay, good. Now I'm unmuted. Okay, thank you. So one thing I'm thinking about at the moment is with COVID-19 and you know, the number of vaccines that will become available in the near future. I think we need to think of you know, how these adverse events will be recorded. You know, there could be projects in terms of social mobilization, you know, trust in the healthcare system, all those kind of things. So I was glad to see this one presentation from Kenya on adverse events following immunization reporting. So if anybody would like to work on something, they can contact me as well. You know, if there's anything in that direction, I will be interested to work on that. Thank you.
Thank you.
Just saying before we go on, Ilse, thanks. Ilse's just informed us that the abstracts are available on the Nelson Mandela website. So that's wonderful. Thanks, Ilse, for that. It's really good.
I could see Felicia raising her hands. She's still there. Oh, I saw her just now, Felicia Williams raising her hands. So now, from what we've said so far, I've jotted down something, and I could find out the groups according to, like Aman suggested. It'd be good to have in groups like antimicrobial resistance and stewardship, medication adherence, NCDs, HIV, TB, looking at adverse drug reactions around that group, and quality indicators, development of quality indicators, and vaccines, pharmacovigilance, and others. So I don't know if Brian has, those are the 5 or so groups I've jotted down.
I think that's enough going forward. I mean, what I'm also doing and be writing to a number of you is a project around medical education. So we had an invitation from Frontiers to produce a paper on, actually it was pre-COVID, about innovation in medical education. So we can turn that around and say, what have been the challenges in Africa, both for pharmacy and medicine, with the COVID-19 and education? So how have we addressed that? So that's another one going forward, but that will just be a one-off project. But I like very much what you're saying, Joseph, and I think, to me, and building on what Hanley said, one of the key areas going forward is what are the unintended consequences. So I know we've discussed that with some of you, but if patients are not able to attend clinics or they're not getting their immunization, then really what are the unintended consequences? So within these groups, I can see that as a research going forward. And I know there are people researching that, but we're the group that have access to most localized data in primary healthcare clinics through our contacts, if you like, in the absence of any formal electronic health records. So maybe that's something going forward we can look at in the various groups. And the other thing, while I'm still speaking and we've got a few minutes towards the end, is is our association with ISPI. So we haven't talked a bit about that, but obviously the Ghana meeting was postponed this year. So how do we carry on with that? And particularly, can we interact with the groups around some of the drug utilization methodology? So that's all I've got to say at the moment.
Okay, I can see Dan's hands up. Dan, I can see your hands up. You have the floor. Okay, maybe it's a bit out of circulation. So that's good, Brian. And so we're adding on something about medical education, pharmacy education, especially in the context of COVID. So now, again, I remember one of the presentations from Kenya, we're talking about use anticoagulants and things like that. So maybe a group around, of course, we have a DUR group that can approach any of these other not commonly discussed topics on medicine utilization, geriatrics and things like that. So we have a group for DUR that we can use, we can work on. So that would be my summary for now. And again, for the quality indicators, Brian. Yeah, okay.
Joseph, can you hear me?
Dan?
Yeah.
Yeah,
I can hear you.
It's also important to include mental health due to the abuse of many psychotropic medicines, codeine, cannabis, you know, as well as the impact of COVID, which has had a lot of led to challenges with mental-related health. So I think that is very important as well.
Okay, okay, great, great. That's very good. Yeah, I think we can add that. So if we succeed in creating the research hub on the website, so we can collate all this into groups, and people that are interested can actually go in and signify their interest, and then that's going to be very very good. Again, another thing that the research collaboration can do for us is mentorship. So, and that is another opportunity that that MURIA gives us, and we've seen examples over the years. You know, very results of good mentorship, productive mentorship. So, and I noticed in this meeting we have a lot of younger colleagues, especially from Kenya and some other South African countries. So it's interesting and it's important that they connect with their senior colleagues using all these links that we are creating, so that things can, they can improve themselves and get some better outcomes from their research. So regarding ISPI, Brian, we're supposed to have a meeting, joint meeting with ISPI in Ghana this year. We couldn't.
Yeah.
Of course, we discussed online and it was postponed until 2021. Let's hope that things will have sorted themselves out with COVID by then, and we will be able to do that. So the relationship with ISPI is also very important. And the manual that you were talking about on drug utilization is actually in collaboration with ISPI. And a lot of guys are also involved in it. So, and apart from that, ISPI has regular webinars on interesting topics on drug utilization and research and pharmacoepidemiology. So it will be good enough if we put it on the site, on the WhatsApp group, for members or other people that are present to connect to these meetings, can benefit a lot from these meetings. So that's another thing that is very important for everybody. Let me see. Brian, any comments?
Yeah, I was thinking you talked about the webinars there, and maybe with Hanley and Ilse there, is how do people join those webinars? Do they, we talked about, I think, when Kwame came to Namibia, about people signing up to ISPI. I think if they're a low-income country, they can get it from a relatively low amount of money each year. So how do people sort of take part in the webinars and help going forward?
I think for the webinars, the important thing is for us to, okay, Anneli.
It was Ilse?
Hello.
That's Ilse.
Ilse, okay.
The webinars at the moment that are presented by ISPE are free of charge, so we just need to sign up. So it is free of charge, and then I know next year there are plans for another series of webinars also. So at the moment, obviously we want people to be part or members of ISPE, but yes, I'm glad that this discussion is taking place between ISPE and MURIA, because we are twins, we are brother and sister. So we need to talk to each other constantly, because we are doing the same thing for the same purpose, because we are passionate researchers in this field. So we've got a lot in common. By the webinars, to the best of my knowledge at the moment, we get the invite, you register free of charge, you attend, and they are high quality. They are organized by ISPE, so it comes from the ISPE head office. Thank you.
Okay, Ilse, obviously I think Joseph has just sent out many emails about this conference to all and sundry. Could you send out an email on the same list, if you like, telling people how they can register going forward?
I will do so. That is a good idea.
Thanks for that. And I think that's wonderful. And I think that shows the value of the collaboration. I mean, through Ilse, we were very lucky, if you like, and then got hold of people who came to Namibia and helped us out, and also to Botswana. So we certainly benefited financially from the ISPE collaboration, as well as academically as well. So we certainly want that to continue.
Thank you. Aman, do you want to say something?
Yeah, I think we always with this MURIA we see a lot of abstracts, but unfortunately not all of them will get published and disseminated in journals. So I think what we could do on that hub on the website is also people can ask for feedback on any draft manuscript or, for example, any grant application. People with experience can provide their feedback before sending to publication. So kind of just helping to improve the publication record of people's within MURIA.
Thank you. That's a good idea. It's brilliant. That's part of the mentoring we're talking about. So it's a good one. So once we sort out the issue on the site, then we'll contact you. We can put us together and put how to script all these things and put them together. Thank you. Any hands raised? Comments? I can't see anyone now. So Brian, if you want to sort of round up.
Okay, well, thanks very much. Sorry, I don't know, is anyone else wanting to talk?
Somebody wants to say something.
I think it's Olianka.
Yes.
Olianka.
Okay, thank you. Sorry, my sincere apologies. My internet went haywire, so I missed out a number of the discussions. Okay, so I'm just wondering if we consider perhaps putting some structure in place for some commitment to also be able to do something for ourselves as a group. I know our colleagues in the institutions, hosting our website have been quite supportive, making use of their facilities, but perhaps, MURIA could also think of hosting independent sites now to be able to also do more than we're being able to do now. I don't know whether we're thinking in this direction, and perhaps we'll also want to consider maybe some token of a kind of membership fee annually to also be able to do, a few things. I don't know whether it's a discussion we want to have now going forward. Just some administrative impetus.
Okay, thank you. If you remember, Brian, in our last meeting in South Africa, I think this was a major discussion. Okay, And it's something that we've never really got around to. I think the general consensus then was that we were to leave things as they were. And again, like Olayinka said, it takes a lot of commitment for things to work. So I don't know. I leave that to you, Brian, and the general. Let's discuss briefly about this.
Thanks for that. I mean, to some extent, I agree with Olayinka, but I'm also very aware. I don't know if Andy Gray's on the phone, but I think when in Namibia, I think Natalie talked about having a constitution and then setting up a formal structure, etc. The problem is, as soon as you start asking for money, you've got to then say, okay, where's your headquarters? And have everything around that in terms of governance and things like that. So that made it very difficult for us going forward because we haven't got that structure in place. And then how do you then work with ISPI? So ISPI, so if people then, do they want to join ISPI Africa? Do they want to join ISPI? Do they want to join us? I think that's the issue that we have. In some ways, as I say, the biggest issue is where will, if we collect money, where will an office be and who will oversee that? Against that, and I know it does collect commitment from a number of us, but we've been very successful in having a loose group, if you like, of interested people. And I keep, I think of, I don't know who ever said in going to war, but it's better to have a few volunteers than many conscripts. And I think that's true, that if people work, and I know there's a group I'm involved in health technology assessment in Europe called UNETA, and that was voluntary, and then they wanted to have it into one formal group and have a single group who then oversees everyone, and that's just folded. So I think our strength is that we do collaborate together. We've got ISPI Africa for a more formal network, so maybe you should get closer to them and help them with administration, and then ISPI generally. So for me, it's more that maybe we carry on with the hubs that we talked about and see how that works. And if we want to more formalize that going forward, then maybe if we go to ISPI and MURIA next year in Africa, in Ghana, then that's the way. But to me, the logistics of collecting money really are problematic. And then is it much better to have, as we have done, a lot of very good people working together, willing to help one another and to go from there. So that's all I've got to say.
Thanks, Brian. If I can come in there, Joseph, I think Brian has raised something that's really important. ISPI has a relatively affordable price for those of us who come from low and middle income countries. They've got all of the infrastructure to collect and to account for that money. Moving money around Africa is not that easy. Not all of our banking systems allow for easy transfer of funds and purchase of Forex. So I would agree with Brian completely. We should, if we want to stay formal, encourage people to join ISPI and to populate the African chapter of ISPI. But in terms of MURIA, let's keep it loose. Let's keep it voluntary. We've done very well up till now. And as long as we can rely on places like Nelson Mandela University with its website, let's continue to go that route rather than spending a lot of time fighting over a constitution and where to put the bank money. I've seen this in FIP as well with the Africa Pharmacy Forum, which has really struggled even to get national organizations to pay across Africa. Thanks, Joseph.
Thank you, Andy. I could see Emily. Emily, your hands are raised. John, could you unmute Emily? Emily? Okay.
Sorry. I don't know. I can't seem to get my hand down. It must be an old hand. I don't see it here on the screen at the moment. Sorry.
Okay.
Handy, why can't we see you in person?
That's our usual style.
Hello.
Hello.
Yeah.
Emily?
Yes.
Okay. Felicia Williams. Okay.
Yes,
Yeah.
I perfectly agree with Andy and Brian to leave things as they are for now, populate a HSP and see how MURA is affiliated with it, and see how things normalize before we think of moving funds around.
Thank you. Thank you very much, Dr. Williams. Brian, I think it seems the consensus is that we should stay as we are, and as much as possible get more people committed. And in actual sense, I want to thank a lot of our colleagues. They are quite committed for the abstracts, for the review of abstracts. I just send them emails, without informing them before, and they were quite, they agreed to review, and within the stipulated time, they sent things back. About 6 of our colleagues. I want to thank Joanita, Professor Boga. I want to thank Sylvia. I want to thank Francis and Olayinka and so many other people that I sent abstracts to. Even I think I sent to Dan in Ghana to review. So people are quite committed, and I think the mere fact that we're able to hold this 2-day meeting as MURIA is a kind of a statement to that effect, that we are quite committed and that we can do these things if we want to get it done. And the summary of it is that I wanted to make a PowerPoint. I forgot that I couldn't make it. I wanted to look at the publications coming out from most of our members before we started MURIA in 2015, and now compare it to the publications that have accumulated by 2020. If we see that, you see a significant change, improvement in that statistics. And that is something that is objective that we can use to say, yes, we are doing something right. And we can get more people involved also. Brian?
Thank you for that, Joseph. I think that's
I think Sam is raising her hand. Sam?
Hello. Yes, hello. My name's Sam. I'm from Brisbane in Australia. Hello, Brian. We met in London, if you may recall.
We did, yeah. Yeah, very much so.
Yeah. So disappointed that we couldn't get to Ghana in July, but looking forward to next July. Just a comment that it's all very Anglophone, MURIA. I'm new to MURIA, so I'm just wondering, you may have had these discussions in previous years, but reaching out to our Francophone brothers and sisters. I was fortunate to connect with Isiaka Sombi and Tene Yamiogo in Burkina Faso. They submitted an abstract, but unfortunately couldn't attend for the presentation yesterday. So I just wondered whether MURIA had made steps in that direction. Thanks.
Maybe I'll answer that, Sam. Thanks very much for that. We haven't deliberately kept out any Francophiles. We've been purposely Pan-African. The only difficulty we've had is here and in our previous MURIA meetings is we got no simultaneous translation. So necessarily everything has to be in English, really. So we haven't deliberately excluded anyone. I mean, I know my boss Lars has worked in Burkina Faso, and I'm also working with people in other West African countries. So the short answer is no, we haven't deliberately excluded anyone, and so the more people from Africa that join us and want to collaborate, then better. So great if we can use your network to expand the MURIA group.
Yeah, and it's possible that when we do have the SB Africa meeting that we can, capitalize on that. We might be able to get more French-speaking people there and develop those links a bit better.
Yeah, and maybe through SB Africa, maybe there might be an opportunity for simultaneous translation because they have more funds than we do, really.
Sure. Thanks.
Thanks.
So Sam, if I may say something, in SB Africa, I think you have regional hubs. And I know for East Africa, I think Sylvia and some other, in South Africa, I think so we have regional hubs. So I think going forward, if we could get some of the colleagues in Burkina Faso, Mali, or anybody interested, we can actually create a Francophone hub for them. And then from there, then you can link up together. So I think it's a good idea if you follow that path.
I like so.
Oh, great. Good idea. Thanks, Joseph.
Okay. So, Brian?
Well, I've actually got very little to say. I thank again very much the Kenyan group, and maybe we can use your facilities in the future if we have any sort of sub-theme workshops around some of the areas that we've discussed. So maybe with Anastasia and John, we can do that. As John said, they're looking to send out people emails with the feedback and then certificates. So if you can fill those in, then our commitment is that we will analyze those and use those going forward, and also put the results into a publication as we've done before. I think the only last thing I've got to say is when we first met, and Andy showed a slide when we first met with Frank Stavilsa in Port Elizabeth in January 2015, where Joseph and Olienka were there and colleagues from Namibia, etc. And Andy, we didn't know if we were going to be successful. And I think this 2 days, and with over 300 participants in most days, shows just how far we've come in quite a short space of time. And Joseph, lastly, Joseph talked about publications, and I think that's very important going forward, particularly for our younger people. My one plea would be that when I look on a number of the university websites, their list of publications from our personnel are very limited. Some of them, and I've looked at, only go as far as 2015-16. So I think it's very important for ourselves and yourselves on our university websites to make sure the publication list is as current as possible, because that shows that we're working together, particularly if we're doing cross-country collaboration. So that is very important that you put those on the websites to show that we are an international group, that we do work with people outside our own university and our own country, and that shows the strength of MURIA going forward. So that's my final plea. Can we all make sure that our university websites for our groups are as up to date as possible? So that's it.
Okay. Okay. So thank you. So we want to thank everybody for taking time to submit their research work, present their research work, listening, and actually taking these 8 hours from these 2 days to be with us. It's been a very fruitful meeting. And of course, we'll keep in touch. We'll get a lot of, your emails address are there. We'll send things around. And then once we create the hubs, either on the WhatsApp group or on the website, we'll let you know. I want to thank Dan for accepting to be the chair of this meeting. And Dan, you are still with us. So I will call on you now to give a brief summary and close the meeting for us. Thank you very much.
Thank you very much, Joseph, and all participants. I can see we still have over 250 participants online, which shows a great interest in medicine utilization research. I'm particularly honored to be made the chair of this MURIA. I remember my first MURIA, I was just a student willing to learn. There have been comments about the quality of the abstracts. It concerns me little because I know those are young people who have to be mentored into senior researchers in medicine utilization. Such is a person like me. I've grown through this platform. So let us be accommodative of junior staff that are coming through, and that's the essence of MURIA, to build capacity in different countries to do medicine utilization research. The ultimate goal, indeed, is quality research. Friends, indeed, good things come to an end. I would say MURIA has been an excellent workshop, and it has come to an end. There are many things we can point out, but let me highlight a few things. One is the birth of a virtual MURIA conference, I think we will never be the same. Our future MURIA meetings will have to incorporate a virtual aspect in order to increase access to such important conferences. By the fact that you see over 250 people are still hooked on, it shows there are gaps in medicine utilization, not only in Africa, but also globally. Allow me to also extend another good thing we can point out are excellent presentations, but I also would appreciate the junior people who have, you know, put their research on the line to be commented on. Students, some of them are students, and they need to be nurtured through the MURIA platform. Again, the networking is one thing to appreciate. I think that's our strength, and let us not split it with monetary constitutions. I think networking is above anything. Until we get at a certain time T where we have enough funds to run ourselves, but I think the networks we have established are something key that have kept us going. And I appreciate that that has come out through these presentations. I would like to thank all the participants. There are over 300, but that will take a lot of time to go through each of these individuals. Allow me to thank the organizers. Of course, if it were not Brian or Olaninka or the MURIA team, Truta, and many others, I don't think this conference would have happened. So on behalf of the organizing committee, let me thank Brian and Olaninka for, you know, setting up this idea that we could have a conference. Secondly, extend my sincere thanks to the Nairobi team. You have set high standards. Personally, I've never seen a conference run like this. And again, I'll go back to my first point, we'll never be the same. We are going to run our conferences much better. We are going to network much better because of the standards that you have set for us. ISP, our partner, I would like to thank you. Many institutions all over the world, I don't want to single out any, including my institution. They are so instrumental as they have given us expertise, reviewers, presenters, mentors, you know. There's so much which is above money that many institutions put in. This conference has had impact on several young people. I can only speak of my institution. Many young people have tuned in and they have benchmarked what other people are doing in Africa and worldwide. I think that is more important to really affirm what you're doing is at par or not at par with what other people are doing. So it goes beyond just presenting abstracts. It also helps to build confidence. Let me conclude from what the previous discussion has directed us into, that we are a big group. And for people to feel part of it, we need to break into groups of interest as suggested. So I think that is the best way forward for people to now carve into the specific groups for medicine utilization that they would like to envisage. However, my call, I think the MURIA group is ready to begin impacting government decisions, ministry decisions. We should take another step now away from publications, just publishing. It's good to publish, but we can make a call through our publications. We can influence decisions in ministries, you know. There are issues around drug abuse. There are issues around, you know, COVID. Through our publications, we can indeed, I believe, I would like to just call maybe the next step, let our research, you know, be applied. Again, while we exit through this MURIA conference, there are still pertinent issues to be considered. During this conference, we have seen there are issues around metrics measuring medicine utilization or reporting. We need to consider how to come up with quality metrics for not only for Africa, but global. I am very confident that this group can challenge themselves to move a step further in validating WHO indicators and making them more suitable for use in Africa rather than just publishing around them. Again, our population in Africa, there are many patient safety issues, leave alone those coming through with COVID. ARVs, it has been well reported in this conference, issues around ARVs, around COVID medicines, around underreporting. It's a call that we need to take another step to speak to those organizations, to speak to the African Union, to present ourselves as a movement that wants to do change rather than to report on the problems that happen. With that, let me thank all of you, especially the organizers, for this wonderful conference and the participants who have remained hooked. Bye-bye for now, and all the best for everyone.
Okay, thank you.
Thank you.
Bye, everyone.
Thanks,
Bye-bye.
Dan. Thanks, Brian. Thanks,
Bye.
Joseph. Bye.
Bye, bye, everyone. Bye, bye, bye.
Thank you.
Bye.
Bye, colleagues.
Bye.
Yeah. So we expect
Hello.
summary. Yeah.
Yes.
Bye for now.
Bye, Margaret.
Yeah.